Sir, सांगा.
Sir, can we start?
Yes, we will start.
Good morning, all of you. I am Professor Gaikwad from the KBC College, head of the department. I welcome you all in this e-conference organized by Department of Chemistry, Karmaveer, Bhaurao, Patil, Vashi. Sir, can we start?
Yes, we will start.
Today we are going to discuss about the drug design and discovery development. On this conference, our quite honorable principal sir, Dr. V.S. Shivankar sir is there. Dr. Shikshan Sahib is there, and Director of Open Division Panvel. I welcome you all of you on behalf of Department of Chemistry and KBC College, Vashi. May I now request our honorable principal sir to give the introductory remark about this conference and the college. Dr. Shikshan Sahib is there, and Director of Open
Namaste. Good morning, all of you. I am Principal Dr. Shivankar. Extend warm welcome to all the dignitaries, Honorable Dr. Sanjay Jagtap, Joint Director, Higher Education, Open Region, Dr. Samir Kulkarni, Dr. Viran Igunpalli, Dr. Deepak Salunkhe, and all the participants on the behalf of all of the staff and students of Karmaveer, Bhaurao, Patil, Vashi. Dear friends, education is one of the best ways for human beings to find his true self. It is also an ideal tool that can be used to define person. For many years, education, even at its primary level, was denied for many in a society due to a multitude of reasons. Friends, times have changed, and still a large portion of the younger generation continue to deprive of quality education. Although the reasons behind this issue have changed, the problem still prevails. To put an end to this dilemma, many primary institutions have opened their doors following the philosophy of education for all. The legendary social reformer and educationalist who adopted and promoted this ideology even before the country got its independence was Padma Bhushan Karmaveer Dr. Bhaurao Patil. By transforming his vision into practice, he founded the Rayat Shikshan Sanstha, which has headquarters at Satara, Maharashtra, in 1909, which has developed into one of the largest educational groups in India, with more than 739 branches, out of which 43 are colleges. The group has permanently transformed the higher education landscape in the state of Maharashtra. One important subsidiary institution of this group is Karmaveer Bhaurao Patil College, Vashi, and it is an elite higher education institution which was established in the year of 1979, with the same mandate of helping the downtrodden and empowering the higher education in the state of Maharashtra. Situated in Vashi, Navi, Mumbai, the college has played a crucial role in offering quality higher education for the prevailing communities in this area, which was established in the year of 1979, based education philosophy. The same mandate of the institute has emerged and empowering the many first generation learners from the community and turned them into professionals who have made a name for themselves across various higher education strata. For the prevailing College, Vashi, which is the first autonomous college in Mumbai, which is accredited by NAC with A plus grade with a CGPA of 3.53 first generation learners. It is also an ISO 9001:2015 who have made a name for themselves, operating with the vision to work for a better tomorrow. The educational advancement of the society while deepening social justice and obligation in mind. The institute cultivates minds and builds characters of young generations by imparting their scientific temperament, organized liability, and human values. of better tomorrow. Dear friends, educational Karmaveer Bhaurao Patil College, first autonomous society, just bestowed upon the prestigious Best College Award by the University of Mumbai. In the field of development and achievement, the Higher Education Forum of India awarded the Certificate of Merit in Innovation to the college. The college has received many prestigious grants from the government of India in the recent past, which includes Best Grant from Department of Science and Technology, Star Scheme Grant and Star Status from Department of Biotechnology, Government of India. Higher Education Component 8 Grant for Certificate of Quality and Excellence from Rashtriya Uchchatar Shiksha Abhiyan MHRD. In the recent past, UGC conferred autonomous status grant to the college in June 2018. The college received star grants last year from UGC. Higher Education Review awarded College of the Year award in 1990. As per the survey conducted by Education World, UGC college ranked 55 in the list of private autonomous colleges in 2018. The college received star congratulate Department of last year from UGC organized that have the potential to become therapeutic agents. The development comprises all the activities involved in transforming from drug candidate to a product approved for marketing by the appropriate regulatory authority. We have invited reputed experts, resource persons who have detailed knowledge about that have the potential to I am very happy and thankful all of them for accepting our invitation. I hope all the participants will get enough knowledge about the theme of this subject. Thank you very much for giving me an opportunity to express my thoughts and welcome all the dignitaries again. Dhanyavad. Thank you. Very happy and thankful all of them for accepting our invitation. I hope all the participants will get enough knowledge about the theme Gaikwad subject.
Gaikwad sir.
Thank you very much for giving me an opportunity
Hello.
to express
Hello.
Yes.
Thank you very much, sir, for valuable the remarks
Backward, sir.
about the conference and inputs about the
Hello.
college and the department. Thank you very much again. I am Mr. Gaikwad, YA, head of the department from the chemistry and the coordinator of this conference. I'm giving a brief introduction
Yes.
to the college.
Thank
Thank you very much, sir, for valuable the remarks
Backward, sir.
about the conference and brief the introduction about the
Hello.
department as well as the college in the conference. Thank you very much again. About the department, Mr. Gaikwad, YA, our department is the biggest department in the coordinator of this conference. 20 brief teaching faculties and 21 teaching faculties. Laboratories and having the introduction about the department reputation in the research area also. We are having multiple research laboratories, recognized research laboratories, department is and more than 500 research papers published during this tenure. And having the most valuable the impact factor as well as the citation. Reputation in the research area also. We have multiple research laboratories, recognized research laboratories, department is more than 500 research papers published during this year. And having the most valuable impact factor as well as the citation. more the funding agencies. We have also very important population center, that MPCB center at our department. This is the prestigious point of our department. This is about the brief introduction about the department. Then I will give you the brief from my point of view. It gives me immense pleasure of happiness in organizing two-day international e-conference on drugs discovery, development, and applications by our department. As the population grows and ages, new areas of medicines need to emerge. The disease in that developing countries are growing increasingly similar to those of the developed one. Time has alarming demand of many new drugs and medical discoveries for many health issues. Identification of lead candidates and their pharmaceutical characteristics. Drug is prolonged journey involving multifaceted aspects for the drug development and regulations. There is the need to develop world-class support and infrastructure for both production and research in Indian pharmaceutical industry to become globally competitive. There is an effort. Drug is there is long journey. There is an drug discovery is an process in which aim at identifying a compound therapeutically useful in world-class treating and cutting a disease. Typically, drug discovery efforts address a biological target that has been shown to play a role in the development of disease to start from the molecule with interesting biological activity. The process of drug discovery involves the identification of API. $1.9 billion. The typical development time for the drug is 10 to 15 years. The development world suffers the major burden of infectious disease, yet the range of the drugs available for the treatment of many infectious diseases is limited. In the past, most drugs have identifying development ingredient from the traditional remedies or by serendipitous discovery. At the present, a new approach is being tried to understand the range of how disease and infections are controlled at molecular and physiological levels and to target the specific entities because of this knowledge. Researchers are interested in investigating carbon nanotubes, scrubbers, and the membranes at the present to separate carbon dioxide being tried to understand plant exhaust. How disease I am highly in-depth chemical college and giving the level, which is encouraging my target working faculties and my students for researchers operation investigating in creating this opportunity. I hope this event will motivate and profit everybody on behalf of
Organizing Committee of the Chemistry Department. I welcome you all of you to Karmaveer Patil College, Vashi, for active participation in the e-conference and making it a grand success. I also like to express my most heartfelt appreciation to all who mention instrumental organization. I welcome you all. Thank you very much. All of you to Karmaveer Patil College, Vashi, for active participation in the e-conference and making it a grand success.
Hello, sir. Patil, madam,
I also like to express my most heartfelt appreciation to all who mention instrumental.
Sir, you introduce Jagtap sir, then Jagtap sir will.
Thank you very much. All of you.
Because about the college, Shivankar sir has finished about the college, so you can proceed.
Hello, sir. Patil, madam,
like to express my most heartfelt appreciation to all mention
Varla sir, you introduce Jagtap sir, then Jagtap sir will.
sir.
About the college, Shivankar sir has finished about the college, so you can proceed.
Yes. Good morning all. I am Dr. E.C. Wadwa, Assistant Professor, KPP College, Vashi, co-convener of this conference. This conference is under the guidance of our principal, Dr. V.S. Shivankar sir, our Joint Director, Dr. Sanjay Jagtap sir, our HOD, Mr. Y. Gaikwad, yes, and Vice Principal, Patil madam. I am Dr. This conference is basically based on drug discovery process and drug development, because we are passing through this COVID-19 and we are going through the importance of the drug discovery. Everybody is thinking about vaccine come, which type of vaccine come, and when this is working. So on the basis of this, to know the knowledge to the students, professors, what is the development process of the drug, how much time is required. So we have organized this conference. This conference is scheduled on 15 and 16 June. Going to this conference, we have said invitation all over the India. From all over India, we have around 1,750 applications. 1,750 participants are there. We have got around 120 research papers. This conference, all research students as well as professors did their recording of paper presentation on the basis of their type of paper presentation, area and nature of paper presentation. Our editorial board from KPP College Vashi and our friends, we have shortlisted around 40 research papers from 120. We have taken only one-third of the research papers. Out of these 40 research papers, we have shortlisted 5 to 6 research paper presentation. This conference is basically on story process editorial board. So I welcome all our students, research shortlisted in this conference, and we have taken only one-third of the research paper. Best luck for this conference, and one best Dr. Sanjay Jagtap, Joint Director Panvel Vision, to give keynote address about this conference. Thank you. This conference is basically on story process.
Thank you, Mr.
I welcome all our students, research
You all.
in this conference, and
I welcome all the participants of this country as well as those participating in this international conference on drug discovery, development and application organized by Karmaveer College, Vashi. Morning, instructor patrons, you all. I welcome all resource persons in this country as well as those participating in international Shivankar, Principal of KPP College, Vashi and application. Dr. Patil, Vice Principal of KPP College, Vashi. Instructor patrons, you all. Gaikwad, Head of the Department, Resource Person, University, as well as Mr. Karmaveer, Principal of KPP, Convener of the Vashi Conference. All the members of the advisory committee and organizing committee. Instructor patrons, you all. Gaikwad, congratulate all the participants of university and the member of the organizing committee. Convener of the within a time, a period of short time, the members of the advisory committee and all the members of the organizing committee have taken the efforts to organize such wonderful thematic conference on drug discovery and development and its application. The organizing committee, and which is a very important aspect in today's scenario. What is the role of the chemistry members of the organizing committee? That is the role of the scientific community. What is the role of the researcher? What is the role of industry conference on chemistry? And how they can make and development and its application sustainable, and which is certain development. Important aspect, sustainable design of the drug, sustainable discovery of the drug, and its application for common man, common people, scientific community. Because our world is segregated into different steps. How they can make can say that the level of the country is sustainable. You can say there is a developing country. You can say there are poor countries. And in such sustainable, diversified world, basically you have to develop such kind of contracts which are affordable because common people, which are affordable to the poor countries, which are affordable to all the common. You can say that the level of the country. What is the role of the chemistry person? What is the role of the scientist? What is the role of the chemistry researchers? Those who have participated, I have learned that from the group with Bhaurao, more than 1,700 participants have registered for this conference. And it is very good efforts of this organizing committee to gather such kind of gathering on these important issues and burning issues for drug discovery and development. What is the role of the chemistry person? How to design the chemical compound? How to design the primary structure? It is very good. Softwares are available. What are three softwares are available? And with the help of this free software or this help of this technological platform, the chemistry people can design the drug and what is their structure, how it can be manufactured, how it can be make it applicable, and what kind of raw materials are required, and whether the technology is available in our country or is available in other countries, and how it can be available. And research and development, what is R&D? You can say that R&D at college level, R&D at research institute level, R&D at industrial level, R&D at universities, R&D at national institutes. So R&D plays a very important role in such kind of discovery. Either it is a drug discovery or either it is other kind of discovery. So discovery is related with the R&D, and the country leads the pioneer in any R&D. Where there R&D is playing important role. And that's why if you take the example of the United States of America, you take the example of the Japan, you take the example of the small country like Singapore, you take the example of Taiwan and China. So those countries are investing more investment in the R&D, and that's why the research in the countries like China, Japan, and America, it is accepted by the entire world, and it is very available in the form of the product. So IPR is one of the important aspects while you are working on the drug design or the discovery. So your intellectual property rights, so filing of the patent or the filing of the international patent or the filing of the Indian patent is one of the important aspects. So we have to take into consideration because the publication is not an issue, but filing the patent and your property, intellectual property rights should be protected. And that's why the IPR is also one of the important aspects. If you see, most of the Indian patents are acquired by the US multinational companies. More than 80% of the Indian patents they were awarded to the US multinational companies. Only 20% patent Indian patents. I'm talking about the international patents. So how the multinational companies playing important role in the R&D as well as in the filing the patents and awarding the patent. So researchers of the Indian continent, they have to think about this IPR and the awareness of the IPR. What is the importance of the IPR? The role of the IPR is protecting rights to the designing of the drug as well as its application and the production. Then one more thing I have to emphasize, that is the collaborative approach. So siloed research are not that much successful. So interdisciplinary research, collaborative research is one of the important aspects in such type of drug design and drug discovery or its application. Collaborative approach within the organization, within the institutions, within the different countries. So collaborative approach must be done. Then important thing, as I said earlier, there is a different status of the countries developing to the poor country, and that's why whatever drug is designed, and the drug design is a cost effective, it is a common man drug, and it is widely used by the human welfare of the human community. So a lot of research is required. One said that more than 15 years or 20 years are required for the and that's why whatever the interference design is the cost effective for the common man and it is widely used by the welfare of the human community. So a lot of research is cut down the the mic of for 15 years or 20 years are required for the and that's why a lot of research is required in the lot of research is required in the area.
What was the mute yourself?
Vadva sir, mute yourself.
Okay, sir.
Lot of research is required in the area in the drug discovery and if you take the Indian perspective, so India is a pioneer member in the area of so many drugs is mostly the generic drug production. So India is a powerful country in the chemicals discovery and drug discovery and lot of companies are working with the multinationals and lot of companies are playing very important role in the drug discovery as well as whatever the raw material is required for the formulations or raw material is required for such type of life-saving drug. So now China is the world leader in the raw material suppliers for the drug industry of the entire globe and including India. Because China, if you take the example of the China, in the drug discovery, China in 1991 and China in 1920. So it is a period of more than 30 or 29 years. So what is the prospective growth of the China? Every respect China develops their industry, China develops the raw materials, China develops the supply chain of entire globe. And even though we are India is one of the leading countries in the drug production, whatever the raw material required, the entire raw material chain to be imported from the China. And it is a very important aspect of the supply chain of the drug. So even India is the leader in the pharmaceutical and generic drug, vaccine production, as well as it depends on the basic raw material. Just I mentioned that China is providing the raw material for the development of the drug. So it is for the raw material, the authenticity and the affordability is the major concern. So whatever the chloroquine drug required for this malaria, and now it is for a certain extent it is used for the cure of the COVID-19. Whatever the raw material required, that raw material is imported from the China, and that's why during the COVID-19, some Chinese companies increase the drug required for this malaria, and now it is I don't understand what's going on. It is for the cure of the COVID-19. Whatever raw material required, that raw material is imported from the China. During the COVID-19, some Chinese companies increase the drug required for this malaria,
might be perhaps
I don't understand what's going on.
biggest encouragement of
some Chinese
सगळ्यांना म्यूट करा.
लाईव्ह स्ट्रीमिंग होतं ना, 1700 लोक बघतात.
biggest encouragement of
So India is the world leader in the generic drug production, as I said earlier, and 20% generic global drug market is dominated by India. It accounts for more than $17.3 billion, and the countries like USA, countries like South Africa, Russia, UK, Brazil, Nigeria, and European Union. So all these countries are importing the drug from the India. So India is very powerful in the drug manufacturing and market. So very important aspect in this conference. I congratulate all these organizers. So see the example, the hydrochloroquine, all these countries used against the malaria, and now it is used against the COVID-19. India is very powerful to some extent. It is useful to the cure of COVID-19. Again, there is a controversy. 2 days back, the World Health Organization, on the basis of some research paper in the eminent medical research paper, they say that the hydrochloroquine is not a proper drug. It is mostly having the side effect, and that's why the WHO again banned that hydrochloroquine. Immediately after 10-15 days, India protests that it is already used for so many diseases, and again WHO that research paper has indicated by that journal, and again the WHO has authorized to certain extent is hydrochloroquine. So hydrochloroquine, we are the pioneers, we are the pioneer member for the drug discovery as well as the production of hydroxychloroquine, and the role of MNC, role of World Health Organization in the biggest market of drug in the entire globe is very important for the framing the policy as well as the drug discovery and its application. A lot of investment is required in R&D, research and development. If you take the example of the COVID-19 pandemic, it was started in the month of December 2019, and still it is very important, spreading vast pace, and more than 85 lakhs people they are affected worldwide, and so many companies and so many universities, Oxford University or Roche company of America, and Japanese companies, Indian companies, Serum Institutes or Institute of Virology, and lot of other biotechnology companies are involved, and more than 100 companies are working on the this COVID-19 drug discovery, vaccine, and they are trying to use the drug against this COVID-19 deadly virus. Some of them they undergo the clinical trials at the human itself in China, as well as in the Britain, as well as in the US. To a certain extent, the second level trials, some third level trials are going on, and let us hope better for the best as well as better. And I will quote some examples. Maybe Karmaveer is a collective drug, a broad spectrum viral drug which inhibits the viral RNA polymerase. It is used against COVID-19 in Japan, and in some extent it can control this example. Then another drug is Remdesivir, another antiviral drug used against COVID-19 as well as the SARS, which inhibits COVID-19 polymerase. American company in Japan is a pioneer with an intellectual property rights, and present in America largely is Remdesivir drug is used for controlling the critical patients of the COVID-19, and worldwide our Indian Director Drug Controller General of India is also allowed to use this Remdesivir for the restricted use in the critical patient, and the government of Maharashtra has also ordered near about the 10,000 vials of this Remdesivir to the American company, and it is a little bit not a top subjective drug, it is a costlier drug because it is not a remedy-free drug. And now the companies like Cipla, Jubilant Life Sciences, Hetero, and Mylon, these four companies, one from India and the rest of the three from the America. So these four companies are trying to get the license from this lead company, which is the pioneer in this production of the remdesivir, and they are starting the production, generic production in our country also. So it is allowed by the DCGI for the restricted and emergency use, not for only it is allowed for the critical patients, and such type of drug which is the pioneer is playing an important role here. Now they are starting it is a very informative and full education going on within 2 days, and all 1,700 participants, researchers for the speaker community, as well as some industry persons, those who have contributed. And I have seen the list of the playing in the importance of the resource person from America, as well as other countries, as well as from other states, and they will give you very good dedication for the stage in this conference. So I declare this e-conference today. E-conference is inaugurated, and I take this opportunity to congratulate all the organizers, participants who are enjoying, and we are going to enjoy 2 days extravaganza for this most preferred subject in the field of chemistry. So my sincere thanks to organizers who have given me the opportunity to give the Q&A address in this e-conference. So once again, I can congratulate all the team of KPP Department of Chemistry, KPP College Vashi, who has taken the pain and who has taken the efforts to organize such wonderful conference on immediate burning issues of drug and discovery and its application. So with this, I can conclude my Q&A address. So thank you very much.
Thanks a lot, sir.
Who has taken the pain and who has taken the efforts to organize such wonderful conference on immediate burning issues of drug and discovery and its application.
Okay, sir.
With this, I can conclude my Q&A address. Thank you very much.
Thanks a lot, sir.
Audible, all of you?
Yes, sir.
Audible? Hello?
Yes, sir.
Yes, you are audible.
Okay, sir.
First, but before that, I will be I want to sir is presently working as line director. Audible, all of you? Panvel region. Audible, and he is a good academic person. He has
Yes, sir.
and he has given more than 80 invited talks.
Shivanka.
Okay, sir.
Yes, sir.
Stop this. It should be at the beginning of the. It should not be.
Okay. So, sir has given detail about the race, and career person, what is the necessary, and what are the role of the industry and the academic, and more elements for the process of the IPL and role of the IIT. So I very much thankful on behalf of the Bhaurao Patil Vashi, the busy schedule, sir has given the time to give the keynote address about this content conference, drug discovery and application. So I again thank the IPL, thank you very much. Thankful on behalf of the Bhaurao Patil Vashi, the busy schedule, sir has given the time to give the keynote address about this conference.
इंट्रोडक्शन देतील ना पाटील मॅडम. हा चालतंय. हो अरे विकास
हा मी देतोय. Dr. Samir Kulkarni sir is on the line, sir.
has given the time.
5 minutes, sir. Sir is coming in 5 minutes. Yes, sir.
Okay, okay. Start the introduction.
Introduction देतील ना. Yes, sir.
Very good morning to all. After an excellent keynote address by Honorable Dr. Jacob sir, on the line, sir. Next session is of Dr. Samir Kulkarni sir.
Sir is coming in 5 minutes.
To introduce the today's speaker, introduction, Dr. Samir Kulkarni sir is presently working as the director of Innovation, Incubation and Linkage National Center for the Nanoscience and the Nanotechnology of the Mumbai University. Is Dr. Samir Kulkarni sir, and the postdoctoral research associate from the Oakridge University National Laboratory, who introduced in November 2001 to 2003, and for developed the biochemical catalyst for enzyme sulfate for the reduction of the desulfatization. Then for the postdoctoral fellow of the University of the Cincinnati from July 2003 to June 2004, for a study the mechanism of the enzyme lysimamide ribonucleotide transformation and for the enzyme catalyzing the third step in the biosynthetic pathway which has been the postdoctoral fellow of the University of the Cincinnati from July 2003 to June 2004, for a study in US Limited Biotechnology Research Laboratory, worked as a senior research scientist from September 2004 to 2007, and then for joined the Dr. Reddy's Laboratory Biologic Development Center, where for a maximum shift here, worked as senior scientist for the document process development from 2007 to 2009, principal scientist in the process development from 2009 to 2012 research laboratory, and then associate director from January 13 to January 2014. After that, for a joined the Biologic Development Center, where for a maximum shift here, 2011, and for a joined the Intax Biopharmaceuticals Ahmedabad from 2007 to work as associate vice president from July 2015 to July 2017. Then for the independent consultant in the biosimilar development, and as I told earlier, presently for working the director Mumbai University for the Innovation, Incubation and Linkage and the National Center for the Nanoscience from February 2019. Such a type of the personality which are the great research in the field of the drug discovery has accepted our invitation. I welcome sir on behalf of Honorable Principal Speaker sir, Department of the Chemistry and the Research Sanstha, Patil. Innovation, and Linkage, feature his knowledge on the metal vector drug challenges and the opportunities. Thank you very much. Such a type of the personality which are the great research in the field of the drug discovery has accepted our invitation. I welcome sir on behalf of Honorable Principal Speaker sir, Department of the Chemistry and the Research Sanstha, Patil. Innovation, and Linkage, for a finish on the metal vector drug challenges and the opportunities. Thank you very much. For Oliver, which are the great research in the field of the drug audible. I welcome sir.
5 minutes Samir Kulkarni sir is joining.
Principal Speaker sir, Department of the Chemistry and the Research Sanstha, Patil. Innovation, and Linkage, for a finish on the metal vector drug challenges and the opportunities. Thank you very much. For Oliver, which are the great research in the field of the audible. I welcome sir.
5 minutes Samir Kulkarni sir is joining.
Principal Speaker sir, Department of the Chemistry and the Research Sanstha, Patil. Innovation, and Linkage, for a finish on the metal vector drug challenges and the opportunities. Thank you very much. For Oliver, which are the great research in the field of the audible. I welcome sir.
5 minutes Samir Kulkarni sir is joining.
Principal Speaker sir, Department of the Chemistry and the Research Sanstha, Patil. Innovation, and Linkage, for a finish on the metal vector drug challenges and the opportunities. Thank you very much. For Oliver, which are the great research in the field of the audible.
I minutes Samir Kulkarni sir is joining.
Principal Speaker sir, Department of the Chemistry and the Research Sanstha, Patil. Innovation, and Linkage, for a finish on the metal vector drug challenges and the opportunities. Thank you very much. For Oliver, which are the great research in the field of
Yes, sir. Kulkarni sir join झाले.
No, just start.
I request Dr. Samir Kulkarni to deliver their important talk on drug discovery. Please, sir. Introduction sir थोडं अगोदर झालं ते. Yes, sir. Kulkarni sir join झाले.
Was a bit tied up in other things.
I request Dr. Samir Kulkarni.
I'll just share my screen.
Yes, sir. There is important talk.
Yeah. Then we can start.
Yes, introduction sir थोडं अगोदर झालं ते.
ऑडिओ सगळ्यांची म्यूट करा.
Kulkarni sir join झाले. केले सर सगळ्यांनी.
Was a bit tied up in other
I
things.
request Dr. Samir माझा मिळत.
I'll just share my screen.
Yes, sir. There is important talk.
Yeah. Then we can start. Hello. I'm sorry.
सर इथे एक ॲरो आहे बघा ॲरो ते एक असा ॲरो बार आहे त्याच्यावरती एक ॲरो
Share my screen.
सिस्को वेबचा शेअर कंटेंट म्हणून ओपन करून ठेवा.
Hello. So taking the genetic fragment from that particular native animal or organism and putting it into E. coli. And similarly, several such biologics drugs have also been developed. Taking the genetic fragment from that particular native animal. What are some of those, these commercial pharmaceutical products? And similarly, the recombinant DNA technology-based products mainly the genetic fragment from that comprise of monoclonal antibodies. These commercial pharmaceutical products. And similarly, there are hormonal drug DNA technology. There are growth factors. I mean, for example, human growth factor or something like PDGA, platelet-derived growth factor. So there are these growth factors. Even insulin is actually type of a growth factor. Insulin-like growth factor, PDGA. Then there are recombinant combination vaccines. So you might have heard that, single vaccine or now many times these particular vaccines are actually these are protein molecule vaccines which actually get tied up together at the genetic level and then they get expressed and the whole molecule. Many times these vaccines are actually then there are peptides which actually get tied up together. There are peptides like and then they get expressed and the whole molecule. These are basically, for example, peptide. Then there are cardiac arrest. I mean, basically, when there is a clot, I mean, pulmonary embolism or something like that. So, bivalirudin or eptifibatide are actually used to resolve that. Then there are fusion proteins. So, for example, there are proteins where part of the protein belongs to a natural fragment like immunoglobulin, like an antibody. And part of it is from a specific receptor. So, I mean, so basically what happens is when you have a immunoglobulin tied up with a receptor, that receptor actually goes and binds to a specific ligand and targets that particular ligand towards disease management. Then there are antibody conjugate drugs. So, for example, nowadays, I guess in the last maybe 5 years or so, 5 or 10 years, not even 10 actually, maybe 5 or 6 years. Antibodies have been tagged with what we call as payload. Meaning what? The antibody is a specific molecule. It goes and binds to a specific antibody. And this particular payload is actually covalently attached to that particular antibody. So when the antibody goes and say, for example, targets a cancer molecule, I mean cancer cell, then this payload is actually a chemotherapy agent which is released on the cancer cell antibody. So this is something which is now, actually has taken a significant amount of prevalence. Then there are other things which you are quite aware of. Cancer cell. Hello. So this is something which is now actually has taken. Then there are other things like vaccines. There are peptides which are made synthetically as well. Then there are plasma fractionation products. I mean nowadays for COVID-19 we are hearing coalescent plasma is being used. Vaccines. Now, there are serious risks in using coalescent plasma as is, because for a patient, from whom the plasma is actually separated, that patient may have some other viruses. He may have something like hepatitis, or he may have something like hepatitis, or something like, say, even HIV. Now, this particular patient can actually, if this particular patient receives blood or a plasma which has, like, this hepatitis or HIV virus into it, then the COVID-19 patient may die because of that, or may get infected because of that. And these particular cases have actually happened in it. So these are, like, certain things where then the fractionation is something which is considered as a better option, which means that from that particular COVID recovered patient, the plasma is taken up, and from that plasma, actually the antibodies are isolated, and just the antibodies are given after virus treatment to a COVID-19 patient. And this is necessarily, like, enabling the appropriate recovery of that particular COVID-19. Then there are these other technologies, or therapies which are now coming up. I mean, every one of us is aware of stem cell therapy, or gene therapy is another one which is actually coming up significantly. So these are, some of these commercial biopharmaceutical products which are nowadays very prevalent in the market. I'm not going to focus on everything because it's impossible to really go into everything in these 40-45 minutes. What I'm necessarily trying to do today in this particular talk is to give an introduction as to how these particular pharmaceutical or biopharmaceutical or biologics products actually get developed in an industrial setting. So here, like, this is just a how these particular biologics got developed. I mean, and what are the differences? So in the first, on the left hand side, you would see aspirin, which is a standard, pharmaceutical drug. And if you look at that, it's like, 180 Dalton molecules. So that's basically a, very tiny molecule. Then, there are these mid-sized molecules like enoxaparin, which is basically a heparin derivative and used as anticoagulant. Then there is insulin, which are about, say, 4,000 to 6,000 Dalton molecules. Then there are these particular molecules like GCSF or human growth hormone. GCSF is granulocyte colony stimulating factors. And these are somewhere in the range of maybe 18 to 25,000. Erythropoietin is about 30,000. Then the other molecules are like monoclonal antibodies, which are basically the huge molecules, about 150,000 Dalton molecules. So, necessarily what it tries to imply is the complexity of these particular products keep on increasing because of the size. And as you might be aware, that proteins are not, active only because of their, structure, or basically only because of their amino acid linking or something. They are active because of their three-dimensional structure. They are active because of their certain shapes and certain, what we call geometry of their structures. And this is something very, very crucial for proteins to necessarily impart that particular biological activity. So some of the indications which are, like, largely managed by biologics today are, like, for example, cancer. So most of these monoclonal antibodies you might have heard about, rituximab, trastuzumab, several of these cetuximab, bevacizumab. Most of these antibodies are basically against the cancer. Now, what it means is cancer cells, depending on the type of cancer, it actually secretes a specific or expresses a specific protein on that particular tumor cell. Now, that protein is targeted by these monoclonal antibodies. So, and monoclonal antibodies are very, very specific to a certain target. So basically what happens is that when these particular targets are identified and these monoclonal antibodies are developed specifically against those protein targets which are expressed on the cancer cells, these monoclonal antibodies go and bind to those specific proteins. And when they bind to those specific proteins, by inducing certain immunogenic responses or certain immunogenic principles in the human cells, it enables the killing of that particular cancer cell. And I will, maybe if we get the time, I'll talk about that, to certain extent in the later part of this particular presentation. Then there are certain, biologics which are used as immunosuppressants. So in cancer, most of these biologics are necessarily enablers, versus in anti-inflammatory kind of application, these are immunosuppressants. So for example, there are these anti-cytokine antibodies like adalimumab. Adalimumab is a specific antibody which binds to tumor necrosis factor. So tumor necrosis factor is a pro-inflammatory cytokine, basically TNF. That is something which gets secreted when there is inflammation. And if there is excessive secretion of this particular cytokines, it leads to excessive inflammation. Like in the case of, say, for example, rheumatoid arthritis, or ankylosing spondylosis, or Crohn's disease, and things like that. So here, these antibodies, or there are also fusion proteins. Now there is a fusion protein called etanercept. Etanercept basically consists of a base of an antibody, and it is joined together by a receptor of the TNF, tumor necrosis factor. So basically, this particular TNF, the receptor which is on top of that antibody in etanercept, it goes and binds to TNF, tumor necrosis factor. And when it binds to tumor necrosis factor, the overall inflammatory impact of that particular on that site is brought down. And in several diseases like rheumatoid arthritis, ankylosing spondylosis, Crohn's disease, psoriasis, many of these diseases, these pro-inflammatory cytokines are released. For example, interleukins, tumor necrosis factor, and things like that. There are several different types of interleukins. So depending on which interleukin is released, that specific antibody can be utilized. Then there are these hormonal drugs, like for example, erythropoietin. Erythropoietin is, like, as you might be aware, it's basically a precursor, or basically a prodrug, not really a prodrug, but mainly something which stimulates RBCs generation, conversion of erythrocytes to, conversion of erythrocytes into the RBCs. And this particular erythropoietin, in case of chronic renal failure, because the erythropoietin is secreted in the renal cortex, necessarily in chronic renal failure, the erythropoietin goes down. And when it goes down, then it ends up creating anemia into a chronic renal failure patient. And therefore, that patient has to be given erythropoietin externally to manage the anemia. So along with dialysis, most of the times erythropoietin shots are also given to such patients. Then erythropoietin is also used in cancer because chemotherapy basically depletes the RBCs. So like more chemo agents, they are basically machine guns, right? They just go around and not just kill the cancer cells, but they kill the normal cells as well. And in many cases, anemia is resultant because of the killing of RBCs. And again, in this case, erythropoietin acts as a supplementary drug along with the chemotherapy agent. Then there are these follicle stimulating hormones and other gonadotropins, which are like, for example, FSH, or human chorionic gonadotropin hormone, or luteinizing hormone. So these are classical gonadotropins, and these are typically used to rectify the reproductive system problems in both male and female patients. So FSH, for example, is typically used to enable the enable the stimulation of the follicle to accept the egg after copulation. So this is something which are these are some of the drugs which are used very predominantly these days, in general therapy or therapeutic indications. Then parathyroid hormone, PTH. So this is something which necessarily enhances the bone development and all, and is used in osteoarthritis and osteoarthritis and similar bone related conditions. Then I just wanted to include some information in the current scenario of COVID-19. Some of the molecules you might have heard about. Actemra is being tested for rheumatoid. Actemra is being tested for COVID-19. What's the principle behind it? So Actemra is basically a drug by Roche Pharmaceuticals, and the actual generic name of Actemra is tocilizumab. Now, tocilizumab is a drug used in rheumatoid arthritis because it binds to interleukin 6. Now, it has been identified that IL-6 or interleukin 6 is something which is secreted in large quantities during the cytokine storm that a COVID-19 patient faces in the acute respiratory distress syndrome. So when the COVID-19 patient, gets into the pneumonic condition and undergoes the ARDS syndrome, at that particular point of time, IL-6 has been noticed to be very largely secreted. So Actemra is being tested to see if this particular IL-6 can be countered, and the COVID-19 patient, the inflammation of the lungs that ARDS COVID-19 patient undergoes, if that can be reversed. So there are some clinical studies which are going on from Roche in this particular direction. Then there is another drug called Alzumab, which is itolizumab. This has been developed, by Biocon in India. And basically this is again, used for plaque psoriasis. Here the mechanism is that it binds to CD6, again a cytokine, which is released by the B and T lymphocytes. And it is expected that when CD6 is actually countered, again in a COVID-19 cytokine storm impact, this particular impact can be reversed by using a drug like Alzumab, itolizumab. Then there are some new antibody research studies which are going on, where people are trying to develop antibodies specifically against the COVID-19 viral protein. For example, the spike protein, as you might have heard, that COVID-19 coronavirus has those spikes on top of this particular shell of that virus. And this spikes is the one which actually goes and binds to ACE2, which is an enzyme, which is expressed in the human cells. And that is how the virus actually infects. So basically, these particular proteins or the nucleocapsid proteins, this is what this particular antibodies are trying to develop against this, so that this virus can be actually neutralized. And quite a significant amount of development has happened in both Italy and Israel. You might have heard in the news that Israel has developed an antibody and all that. This is what it is. So unfortunately, the news agencies have been reporting this incorrectly. Stating that a vaccine has been developed. This is not a vaccine. This is actually a drug against that particular virus. Now, vaccine is a different story. Vaccine is necessarily like something which is given to a patient, and the memory cells are actually triggered. So B cells, which can either generate basically antigen-specific antibodies. Or it can, some of these B cells actually differentiate into creating memory cells. So in the long term, if there is any future attack of the same antigen, then these memory cells immediately release antibodies which are specific to that antigen, and that particular thing is taken care of. So these molecules which are developed by, say, Israel or Italy, they are not vaccines, but they are actually drugs for this. Now let us look at the commercial opportunity that biologics actually is presenting today in the world. These are some of the reports that I extracted from the internet. And, if you look at the global biologics market, by 2026, it is expected to hit somewhere about $362 billion at a CAGR of about 3%. Now, many times when I speak in conferences or something, I get blank faces, and there is not really excitement when they hear the word, some 300 or 200 or 250 or 500 billion dollars. Now, this is something, probably the reason is because for us, we are not able to really contemplate what does it mean by $1 billion. And therefore, I tend to give that simple piece of mathematics to people, that what does it mean by $1 billion? $1 billion is 7,000 crores. 7,000 crore rupees if you consider 70 rupees at the exchange value. Now, imagine $362 billion global business, right? It is basically a serious amount of business that actually is taking place on these biologics drugs. And today in 2020 itself, we have hit somewhere around 150 to 200 billion dollars. And it is expected in the next 5 to 6 years, it is going to run, jump in by another 150 to 170 billion dollars and reach somewhere close to 360. So this is a huge, huge market. And obviously, wherever there is market, there are efforts in terms of getting people trained in that specific domain, getting people accustomed to that particular subject. And that is what I meant when I said, biotechnology, one of the biggest applications that biotechnology has currently found is in the domain of biologics or biopharmaceuticals. Now, this other, slide, the other chart actually lists out some of these, biomolecules which are in the market currently. So, Keytruda is actually a drug it is called pembrolizumab. It is a drug by Merck. And if you look at the estimation, it is expected to hit $22 billion by 2025. One molecule, okay, one drug is expected to create business of $22 billion by 2025. Now imagine the entire this $22 billion is something like 4 or 5 Dr. Reddy's businesses put together. Okay, just to realize the magnitude of this number. One molecule is equivalent business to the entire business of 4 or 5 Dr. Reddy's put together. Because Dr. Reddy and Dr. 50 molecules which they are putting into the generics market. So basically, there is no wonder or there is no surprise that many of the organizations are actually moving into trying to develop these biologics products. Many pharmaceutical organizations are actually moving in that direction. Then most of these drugs, the least here is somewhere about $7 billion. In fact, in 2020, a drug called adalimumab, just last year, 2019, a drug called adalimumab has done a business of $20 billion. This is Humira, this was by AbbVie. And, you can imagine, again, $20 billion is, equivalent to 4 Dr. Reddy's put together, or maybe 4 or 5 Sun Pharmas put together. So that's the magnitude of business of one molecule. And now there are in tens and hundreds of biologics are being worked across the world in trying to develop them for specific indications. Now, what is the reason? Why are these molecules so, readily accepted by the adverse effects are very minimal? For example, when you work with a monoclonal antibody, it goes and targets specifically a cancer cell. It is not like a chemo agent, which is actually going ahead and just shooting all the cells, normal or cancerous, in one shot. Biologics are basically drugs which go and specifically target cancer cells. So the patient's quality of life is much, much better as compared to that particular patient who is actually taking these chemo agents. So this is why now obviously there are serious challenges in this particular biologics. Like I showed you earlier that image. These are very large molecules, so they are very, very complex to develop. Then there are large incubation periods. Starting from identifying a target to developing a specific monoclonal antibody, to develop a process for it, to characterize that monoclonal antibody. All of that takes very large amount of time. It is much more than a normal conventional pharmaceutical molecule. Because in a conventional pharmaceutical molecule, it is very easy to deduce the structure of that molecule. You do a couple of NMRs, IRs, and all, and get that particular structure very, mass specs and all, and you get the structures very easily. But biologics is not that specific case, because here it is not only the chemical structure of that molecule, but also the shape, size, and everything that matters. And these are certain things which necessarily so for example, you might have heard X-ray diffraction studies on a certain protein and all that. Now these are basically X-ray diffraction studies are actually giving the crystal structure of that particular molecule, determining the three-dimensional structure of this particular. So these are things which are additional over and above what this particular biologics, over and above the pharmaceutical molecule. When I say pharmaceutical, these are basically the low molecular weight, products which are very heavily these days used, right? So in this particular, then another major difference is the clinical evaluation is very different. So in clinical evaluation, along with the safety and efficacy, you also have to do something called immunogenicity, because you are injecting proteins into the patient. These proteins themselves are immunogenic in nature. and the patient can actually respond in a very dramatic immune reaction to a certain biomolecule, and it can actually, because this is a foreign protein which is going into the human cell, and the patient can react very dramatically on the from the from the immunogenicity point of view, and therefore immunogenicity is a very critical evaluation that has to be conducted in these biologics clinical trials. Like I said, opportunity is humongous. I mean, we expected to hit somewhere about $350 billion by 2025. Usually, these molecules are irreplaceable. I mean, when we say hormonal deficiencies, then those hormonal deficiencies can be treated only by these hormones, right? I mean, insulin or erythropoietin or things like that. You can't really use something else. I mean, in case of insulin-dependent diabetic, you have to give insulin. You cannot really go for something else unless people come up with these mimics, the insulin mimics or something like that, which has similar activity like insulin. So far, that has not really happened. Then, say for example, if there is a genetic condition, hereditary issue, then it is only curable by gene therapy. You cannot really go by something which is not really in line. I mean, at the most, symptomatic relief can be brought in, but if you want to cure the disease, it has to go to the root of that particular disease. And most of the time, these particular roots are cured only through biotherapies. Like I mentioned earlier, these are targeted therapies, so therefore they provide very high level of accuracy, and the incidence of side effects are very, very less in this particular products or in this particular line of therapies. Now let us look at the typical biologics development path. So like I said, let's take the example of trying to develop a drug against a specific situation in a patient. Let's say that we want to develop a drug against cancer, and in fact I have an example down the line, but I'll just run early, with this example even now, and we can perhaps keep it at that point of time. So here basically what it does is, so let's say that in breast cancer, okay, in breast cancer, the tumor cells express a specific receptor called HER2 on those particular cells. I mean, on the surface of those cells. Now, what has happened is that HER2 was identified, and then a specific antibody was developed against the HER2. Now, as you know, antibodies are very specific towards the antigens that they are targeting. So HER2 actually was identified as an antigen, and then that particular antibody was developed. This antibody is trastuzumab. Then once that antibody is developed, then it undergoes the discovery research, which is basically trying to understand the mechanism of action of that particular antibody. You have to understand whether it is binding appropriately, whether there are appropriate, whether it is actually inhibiting that particular cell from growing beyond. So, there multiple candidates are actually selected. Then from that candidate, one candidate or two candidates are identified. Okay, these actually are giving me the desired response. So what is the efficiency of binding, and everything has to be good. And from that, once the drug candidate is identified, it is then tested much more explicitly and much more extensively for the functionality. So, for example, what is the binding, whether I am getting the cancer cell death because of this binding, all these aspects. So that is necessarily the biological characteristic, which is actually carried out. The mechanism of action is identified, and that particular whole thing is looked at. Then parallelly, one also has to look into what is the structure of this molecule. So that is where the molecular characterization comes in place. So what are the amino acid sequences of this particular antibody? Then where are the disulfide linkages in this protein? What is the glycosylation in this particular protein? All these, and then what is the impact of that structure on the activity of that particular protein? So what we commonly know as structure activity relationship. But remember, structure activity relationship for a molecule which is say 200 Dalton or 300 Dalton versus a molecule which is 150,000 Dalton is a much more complex determination to really make, and therefore that particular thing usually costs serious amount of time. So this is something which, but nevertheless you have to do it, because you will have to actually present this information to the regulatory agencies when you want to get the approval for your drug for marketing it for a certain indication. So these particular activities are carried out. From here, then it goes into further developing the analytical methodologies which are going to evaluate these particular structural features or the biological features of that. So you have to develop, for example, a bioassay. You have to develop the HPLCs which are going to give you the purity, which are going to identify the structural analogs, which are going to identify which is that analog which is actually responsible for the activity, and which are the impurity analogs, because there can be isomeric forms of such large molecules. So all these particular things have to be identified in the analytical scheme of things, and that is necessarily collectively it is known as something called as characterization, molecular characterization. And once that is done, so both molecular and biological characterization result into identifying certain analytical methodologies which will be utilized for the release of every batch. Then there would be certain analytical methodologies which would be identified as stability indicating parameters or stability indicating assays. Then there are certain methodologies which would actually work as functional assays. Then there are certain methodologies which would actually work as the physicochemical assessment assays. Like for example, in certain cases, you may have to determine through, say, for example, a size exclusion chromatography. You may have to determine this thing, what you call the size variants or the aggregates into that particular molecule. So all of these particular factors actually come together to create a complete analytical package. And then parallelly, you have to actually develop the process for it. So basically, that there is a certain cell which is necessarily cloned with a certain genetic fragment that which necessarily expresses the protein of interest or the antibody. And then that particular cell has to be then cultivated into a fermenter or into a cell culture reactor to a certain volume, and thereby extracted a significant amount of protein from that. Then that protein has to be purified. So the downstream process, the purification scheme, has to be identified. And all of these have to be done in a certain way that this can be scalable. That you can, whatever you are doing at, say, for example, 1 liter or 5 liter, it should be possible to replicate that 1 or 5 liter at something like 500 or 5,000. And that is something which is very, very necessary when you actually work on the research and development side of this program. Now, this is the process and research and development side. So there are these 2-3 different phases. One is the drug discovery, research and development. Then there is the analytical research and development. Then there is the process research and development. These are all different formats of the research and development. And then there are the animal studies, which is again component of the research and development. And then there are clinical evaluations, which is necessary in the phase 1, phase 2, phase 3s and all. And here, as I said, one of the most crucial differences that you to actually carry out immunogenicity study as part of your phase 3. So this is something which is extremely important to understand when we say that, okay, I want to develop a biologics. I want to develop a biomolecule or a biopharmaceutical. Now, this is just I just thought I'll give a flavor of what is the process development really consists of. So like I said, there is a certain genetic fragment that has been identified. That genetic fragment is then taken, put into a specific vector, and that vector is then put into an appropriate expression host system. Like it can be a prokaryotic or a eukaryotic cell. And once it is put into that, then the clone is selected, meaning you will have several different cells which have these particular vectors. And you have to select out that particular cell which is expressing your protein to the maximum quantity and giving maximum activity. And once you get that particular cell, then you take it into your fermentation and develop the fermentation process, fermentation of the cell culture process, where you identify what should be the media, what should be the agitation given, stirring that is to be given, what should be the oxygen supply given to that particular, to ensure that the cells are growing to the maximum extent. So all these optimizations have to be done at the fermentation stage. Then comes the purification. So once you have generated that particular, express that protein and you are getting a lot of protein in the fermenter, then you have to purify. Because these proteins, a fermenter can have millions of, other components or other constituents. And from those millions, you have to take out just one, which is your protein. And therefore, you have to actually identify an appropriate purification scheme which necessarily clears out all the other junk and just brings out your protein. And then once you have that purification scheme, then you get into an appropriate buffering combination, excipients and everything. And then it is basically formulated. And while you do all of this, it necessarily is like, at every single stage, you have to involve appropriate analytical methodologies to evaluate whether I am getting the right protein, whether my process is the right process, whether it is generating the right quantity and quality of the protein. And this is all going to be answered by the analytical methodology. Right. So now, as I have been telling, so biosimilars is something which is, in a way, these are similar to the generics, what we know today as pharmaceuticals and then generics, right? Biosimilars are the generic versions of the novel biologics. Now, the reason for for developing biosimilars is because regulations have understood that to bring out a biologic molecule into the market, there is serious amount of cost implication, and to counter that cost, there is basically most of these drugs are priced very very heavily. I mean, to give some of the examples, when trastuzumab was launched in the US, one therapy cycle used to cost $30,000. When pertuzumab, which is another such molecule that was launched, it used to cost $90,000. Rituximab, before Dr. Reddy's, when we developed this particular molecule in Dr. Reddy's, before that, it used to sell at 2 lakhs rupees per vial. And when Dr. Reddy's launched a biosimilar, we started selling it at 40,000. So basically, that was the tremendous difference that was brought about in reducing the price of that particular drug. And therefore, biosimilars are being encouraged now significantly. And the way they are encouraged is the clinical study has been reduced. So you don't really have to do complete phase 1, phase 2, phase 3 for biosimilars. All you need to really do is pharmacokinetics, which is necessarily bioavailability. And you have to carry out the what you call as safety efficacy study along with the immunogenicity. So this is how, in normal situation where you need, for a novel molecule, somewhere close to 2,000-3,000 patients, maybe more, to determine every single aspect of that particular molecule because it's a novel molecule. In a biosimilar, these particular numbers can be brought down to somewhere around 300-400 or 500 patients. And then, certain very specific observations are made because a lot of information is already known about this particular molecule because there is an innovator molecule which is already existing in the market. However, the only thing is, generics, these particular products can be launched into the markets only after the patents expire. So, and usually the patent life is about, not usually, but the patent life is around 20 years. So unless and until the patent has expired, you are not able to enter that specific domain if there is a pre-existing patent on that molecule. So this is what is necessary for biosimilar business which is coming up. Now, biosimilars business itself is expected to hit somewhere around $100-150 billion by around 2025. Because several molecules are going off patent between, say, 2019 to 2025. And by 2030, it probably would hit somewhere around $250 billion. So this is some serious amount of business opportunity that is in the waiting. And India is mainly, most of the organizations in India are targeting biosimilars. And this is just a brief definition of what biosimilar is defined as by the regulatory agency. It is necessarily known as a highly similar molecule, notwithstanding minor differences in clinically inactive components. What it means is that basically a biosimilar is a very similar, structurally very similar molecule like the originator molecule. But certain differences are permitted as long as those differences do not come together in creating any kind of problems at the clinical side. So there should not be any unknown safety issues or additional safety issues, or the immunogenicity should not be very different than what the original molecule is actually doing. So these are some of the, and of course, the efficacy wise, it has to be non-inferior. So it doesn't have to be a superior or even an equivalent, but it has to be a non-inferior. In other words, it has to be at least equivalent. So this is these are some of the requirements as defined by the regulatory agencies for a biosimilar. Now, when we talk about similar, then what are those similarities? So similarities are necessarily determined at structural level, at the preclinical or animal toxicity level, and the clinical level. meaning the clinical safety and efficacy, like I said. So these are like some of the typical functions in a during biologics development. I have more or less already touched upon them, so I'm not going to get into too many details. Maybe just a few in this. So like I said, research and development, it consists mainly of target identification, candidate generation, and then the identifying the development path, the scale of operation, the market demographies, or which markets do you want to really access with this drug, where is the disease indication mainly there, and in which country. So I want to probably target that country. So then for that country, what are the regulatory requirements? So I probably need to bring in those specifics into my development plan. Then the process and analytical development, like I was telling you, the upstream and downstream, or the fermentation and the purification processes and all. Then your formulation development, what are the excipients, what is the stability going to be impacted by? So all these particular things. Then the animal studies, in terms of is there any tissue cross-reactivity? Is there any unnatural adverse event in animals? What are the abnormal toxicities? What are the chronic toxicities? All of these have to be actually then the clinical development, which is necessarily looking at initially the safety as phase 1, then identifying the dose and everything, and the pharmacokinetics, and then eventually safety and efficacy. And then finally, once the product is launched, one has to go into the pharmacovigilance. And yes, somebody has a question. I'm not able to hear. The voice is distorted. I'm sorry. You can put down the question in the chat, and once I'm done with the presentation, we can take up the questions one after. Okay, so should I continue? All right, so so the next thing is about manufacturing. Now in manufacturing, basically one of the since you're working on biopharmaceuticals and you're working on the cell culture media and everything. Obviously, this is a very rich medium for microbial contamination and things. Therefore, there are stringent environmental and process conditions which are imposed on the during the production of this particular molecule. So these are always carried out in classified environment. So you might have heard about this clean room concepts. So these are class 100,000, class 10,000, these sort of things. Then the formulation and the filling is done at almost something around class 1000 to class 100. So under a HEPA, basically, laminar flow. So these are some of the things which are very important. These are so GMP factors in these are to be monitored very, very carefully. And even regulatory agencies. So after you have, say, for example, applied for a certain drug with a regulatory agency, it is not sufficient to simply justify the science behind that drug. It is equally important to demonstrate that you are capable of producing that drug in an absolutely clean and GMP-based environment, and then basically are following that particular practice batch after batch. And this is something which is actually audited. This is something which is actually examined by the regulatory inspectors. They come into your sites, they take a look at how you are going to manufacture, whether you have all the processes, whether you have all the SOPs listed out, whether you have documented all your research, whether you have actually demonstrated what are the control parameters in your processes, and how are you going to manage those control parameters, whether you have put down protocols for managing any sort of deviations from what you expect that the process should really go as. All these factors are necessarily to be looked at, and any deviation has to essentially come in with a very immediate and effective solution, troubleshooting tool. So this is something which is very, very important in manufacturing of these biomolecules. And obviously, in almost all the parenterals, it is more or less similar, because when we say parenteral or an intravenous or subcutaneous injection, then you are directly incorporating that into a human being. And in those stages, when you are putting something in intravenous route of administration, you have to be obviously extra careful. However, in biologics case, this particular thing is several notches above, because the molecule that you are dealing with has serious amount of propensity to interact with any kind of contaminants that can come in. And to give an example, there was a huge case or a very significant side effect observed in erythropoietin sometime in maybe 2002, 3 or that sort of timeline. And what was observed was that there several patients were suffering from something called as PRC. And so this was basically a kind of aplasia, mottling of the skin color and things like that. And this was basically because the hemoglobin that was actually getting generated was some way of a different nature. Now it was observed that erythropoietin was interacting with the rubber chemicals from the rubber, which were used for the cap of that particular vial. And that rubber was leaching out into the solution, and erythropoietin was interacting with that rubber and creating a different molecule, which was resulting into a very dramatic adverse effect. So this was what was identified, and then after that, all the biomolecules, when they are putting into any kind of a injection vial, the rubber caps have to be Teflon coated to avoid any kind of leaching into this particular thing. So these are very common, or these are very susceptibilities. These are susceptibilities that have been identified in this kind of biomolecules, because there are several functional groups. You can imagine, right? A protein is made up of a combination of 20 different amino acids, and there are several functional groups, several side chains of amino acids. There are sulfhydryls, there are hydroxyls, there are amines, and a lot of these functional opportunities are there for different types of reactions to take place. I mean, there can be deamidation, there can be oxidation, there can be isomerization. So a lot of these structural changes can happen in a protein molecule, and therefore it is very important to ensure that the molecule is given appropriate environment throughout its lifetime. So not just during its manufacturing, but even during its shelf life. That there is if many a times you have to actually purge the vial with nitrogen to avoid oxygen in in the headspace of it, because oxygen can be reactive with time. So these are certain things which have to be looked at during the R&D, and this information has to be passed during manufacturing, and then ensure that the product is manufactured and packed in an appropriate way. And analytics, as I mentioned, so the goals are to identify and characterize the product and establish its purity. So there are several of these analytical techniques which are used, like spectroscopic techniques, for example, mass, NMR, IR, then circular dichroism, which actually gives you the secondary structure. Then the DNA and protein sequencing that necessarily has to come in. Then electrophoresis, which is necessarily separating the proteins on the basis of either charge or size. Then biological functionality, like I said, whether the protein is actually creating that particular action or is capable of creating that action has to be tested out in an in vitro manner, where the cancer cells are actually grown in the lab, and then you test out whether you are able to get cytotoxicity. Whether you are able to kill the cancer cells. Dr. Wadhwa, how much time do I have? Hello? Hello? Okay, so sorry? No, how much time do I have?
Hello, sir.
Yeah.
Sir, you take your own time, sir. No problem, sir. No problem.
Okay.
Okay.
All right. So, probably another maybe 10 to 15 minutes.
Yes, sir.
Anyway, so basically, once these analytical methodologies are developed, then you have to transfer these methodologies from R&D to the quality control labs, where every batch would be tested by these analytical methods and necessarily released into the market. And these particular things have to be assured or have to be kept as a check. All the production methods and the production practices, the analytical methods and the analytical practices, all of them have to be checked appropriately by a department called, or a team of people who are carrying out the quality assurance and the validation job. in an organization. So basically, quality assurance is somewhere where you are dealing with, since you are dealing with highly susceptible molecules, you have to always be aware about the microbial and viral contamination of at different stages. And a quality assurance person has to ensure that they are monitoring the processes appropriately in an organization to ensure that basically this particular practices or the SOPs are being followed appropriately. SOPs are actually put together in place, and people are following those SOPs appropriately. Because this is something which is extremely important and is going to be monitored very crucially by the regulatory agencies. And then regulatory, regulatory department in an organization, it necessarily has to, ensure that the dossiers are created in accordance. I mean, dossiers in the sense like, basically a bunch of documents that have to be put together in accordance with what the different regulatory agencies across the different countries actually require. And these are the people who actually interact very intimately or closely with the research and development teams, and necessarily collect information appropriately in accordance with what is required to the different agencies. And then in terms of the preclinical or the clinical development, this is of course sequential. So initially one has to undergo extensive animal studies. In case of novel molecules, animal studies are very, very extensive. You have to identify different, including the pharmacokinetics in animals, including the efficacy in animals, and for that you need an appropriate animal model. So you need, for example, if this is an anti-cancer drug, you need a appropriate animal with that specific cancer, so that you are able to test out your drug. Then the same thing, when it gets into the clinical domain, you have to identify again, in patients, you have to identify what is the half-life of the molecule, what is the efficacy profile of that particular molecule, what is the safety profile of that molecule. And in case of biologics, as I mentioned, it has to also be immunogenicity of that. So these are some of the very crucial requirements which have to be determined for a biomolecule or a biopharmaceutical development. Now, this is just an example. I thought I'll cover how many slides I have, but hopefully, I can cover some of these. So basically, this is just an example of how do you develop an anti-cancer drug. So here, like I said, a target has to be identified. Now, a receptor, basically, that is overexpressed on a cancer cell, or receptor is necessarily some protein that gets overexpressed on a cancer cell. Now, here I've taken two examples of CD20 and HER2. So CD20 is a protein that gets expressed on non-Hodgkin's lymphoma cells. So non-Hodgkin's lymphoma is a type of cancer, blood cancer, and it gets expressed on these particular cells. And this particular CD20, there is a specific antibody that has been developed for CD20, which is rituximab, and it goes and binds to CD20 specifically. And we will look at what is the mechanism of action there. Then there are certain other types of drugs like growth factor that promotes tumor growth. So basically, there are antibodies which are targeting these growth factors. For example, vascular endothelial growth factor, VEGF, or epidermal growth factor. So VEGF, so there is an antibody called bevacizumab, which specifically binds to VEGF. Now, what happens is once the antibody binds to VEGF, it actually takes that particular VEGF out of the equation. And VEGF is responsible to create blood supply or to create the vasculature into a tumor. So once the blood supply is stopped, the tumor automatically reduces or dies out. And the same is the case with epidermal growth factor, where basically the growth, multiplication of the cells is inhibited by targeting the epidermal growth factor. So there is an antibody called cetuximab, which specifically targets epidermal growth factor. So these are the different mechanisms which are used. And then in rheumatoid arthritis, basically, like I mentioned, there are these pro-inflammatory cytokines like tumor necrosis factor and interleukins, and these are antibodies developed against these cytokines. Now, taking the example of rituximab, it binds to the receptor called CD20, or a protein which is expressed in this non-Hodgkin's lymphomatic cells. And this CD20, which is overexpressed, once it is bound, rituximab necessarily kills that cancer cell by 3 different mechanisms. These are called antibody-dependent cell-mediated cytotoxicity, complement-dependent cytotoxicity or CDC, and apoptosis. Now, what are these different mechanisms? So this is rituximab, and that's the CD20, which is necessarily say on a non-Hodgkin lymphoma cell. Now, when rituximab goes and binds specifically to that CD20, and once it is bound to that CD20, here basically what it does is from the FC region. So for a antibody, this is called as the FAB region. The Y, the V shape in that Y-shaped antibody is the FAB region, which is actually responsible to bind to a certain antigen. And then the tail of that is called the FC region. It's basically a constant region. And from the tail, it actually recruits a natural killer cell. So from the human, immune systems, these natural killer cells are recruited by rituximab, and these natural killer cells then come in and they kill this particular cancer cell. That is necessarily known as antibody-dependent cell-mediated cytotoxicity or ADCC. Then there is another mechanism called complement-dependent cytotoxicity. What it does is, again, rituximab binds to CD20. And once it is bound to CD20, it triggers a mechanism of different proteins to actually interact, starting from the C1q protein. And these proteins interact and they create a membrane attack complex. And this membrane attack complex goes and sits on that cancer cell and basically punctures that cell, and thereby brings about the death of that particular cell. So as you can see, without the CD20, there is no action. Therefore, unless and until CD20 is expressed, and therefore, since CD20 is expressed mainly on the cancer cells, rituximab acts only on the cancer cells, and therefore the side effects are very, very minimal. Then the third mechanism is apoptosis. So once this particular rituximab binds to that CD20, the further, the dimerization of these CD20s is avoided, and this dimerization is required for triggering mechanisms for the multiplication of this particular cancer cell. And this particular multiplication, once it is avoided, eventually the cell death just takes place by itself, and that is necessary apoptosis. So these are specific mechanisms which are brought about by a antibody. And since, like I said, they are so specific to a cancer cell, the side effects of these antibodies are very limited. And that is one of the primary reasons why these therapies are much more acceptable or much more accepted over chemotherapies. Now, Indian industry, mainly we are all working on biosimilars, except maybe some exceptions, like for example, Biocon actually developed that particular molecule. But as much as I am aware, I think they developed it in partnership with some other major pharma. And basically, why do we have to work on biosimilars? Because obviously, as a country, as a developing country or as a country which has just crossed certain economic barriers, we still need low-cost options. I mean, how many of us can afford something like a therapy cycle of 2 lakhs rupees, one injection of 2 lakhs rupees? It is not really. And if you need, say, 6 injections for a certain therapy, I mean, there itself you need 12 lakhs rupees, right? How many of us have that kind of monetary strength? So these are certain things for which biosimilars development has been encouraged in the country, and most of the organizations in India are actually trying to work on biosimilars. The technology know-how in the country is building up rapidly because there are many people who have either returned from the Western countries and after working in some of these organizations, the global organizations, and they are necessarily helping out the Indian organizations to build up quality drugs, quality biosimilars. Our regulatory awareness is also improving drastically. Earlier, our guidelines were very vague and they were very easy to manage, but now we are more or less come up with guidelines which are at par with the global requirements. And the main reason for that is because if you don't have guidelines which are going to enable standard quality drugs, then you would end up with getting drugs which will probably get approved in India, but we will not be able to bring it to global. And under that particular thought, the government has actually ensured that the guidelines are of appropriate nature or of appropriate, scrutiny. That basically, good quality drugs, good quality biosimilars are getting approved in India. And India, which is known as the pharmacy of the world, we are appropriately, staged to necessarily make it big even on the biosimilars. Some of the examples in terms of the kind of cost reduction that has been brought about. Now, this is an old slide. I'm sure these numbers are still further down now. Basically, filgrastim, that is the GCS, Goldman Sachs correlation factor, the value of that drug has been brought down by 83%. So where earlier this drug used to be sold at somewhere about 12,000 or 14,000 rupees one injection, is now being sold for about 100,000 or 1200, even lesser. Pegfilgrastim by about 57%, rituximab about 60%, teriparatide, that is PTH, is about 70%, etanercept is 72%, ranibizumab, which is basically Lucentis, which is used in age-related diabetic macular edema in the eyes. This particular drug has been brought down by about 40%, and it is only going down. So, as I said, this is an old slide. Hopefully, now these numbers are still better. So that is what I thought I would cover, as part of this particular talk today. Just as I said, wanted to give you a flavor of what is biologics, what are biologics, what does it really include as developing biomolecules or biopharmaceuticals, and what kind of complications are there. Because to be honest, I do not think that this is anybody's cup of tea. I mean, unless and until you know the subject very well, unless and until you know, you have good good financial strength to support these programs, it is extremely futile to get into this kind of development businesses. And you might have seen me speaking mainly from the business point of view, of course, technical and business, because that has been my background. I mean, I have been working in the industry for the last 20 years. It is only last year that I have joined the university, and basically I have been involved in developing these biopharmaceuticals in different organizations within India, and even before that in the US. So thank you very much for your time and attention. If there are any questions, I would be happy to take.
Any questions?
There are some chat messages. Let me just
Nobody has answered.
No, I mean, I had actually mentioned that you can put down the questions on chat.
They are.
If there are any questions on the chat, I just look at that.
Yes, sir. Thakur sir. Thakur sir.
हाँ.
Yes, sir.
हाँ. येतोय आवाज माझा?
Yes, sir.
आवाज येतो ना? Okay. Friends, just you listen to respected Dr. Sambir sir, the former director, Innovation, Incubation and Linkage, and the National Center for Nanoscience and Nanotechnology, the University of Mumbai, in this 2 days international conference of the drug discovery, development, and the applications. As I told in the introduction, starting from the 2001, recently is in this field, and I will say that sir has opened the panorama box of the biologics in front of us. Starting with what is the basic knowledge. Sir has given the applications of the biotechnology. Sir has given the insight about the commercial biopharmaceutical products, recombinant the technology, and then major indications, and this about the cancer, immunosuppressants, and the hormones. And the whole world is suffering from this COVID-19 pandemics, given the knowledge about the molecular identified, tested against the COVID-19, like the Actemra, Aljon, new antibody research. In his session, sir has given the many different opportunities in the biologics, global biologics market, focus on the top 10 best-selling drugs. They're about in the year 2025, the global data. Then, sir also give the knowledge about the entry barriers for the biologics as well as the opportunities which are there. We have given the typical novel plan about the biological development, different formats of the research and the development, biopharmaceutical processes and the development. We have opened the world of the biosimilars, as given by the define the regulatory agencies, similarity considerations, functions in the biological development, in a research and the development, manufacturing, analytical, quality assurance, process, and the validation. We also give the knowledge about the preclinical and the clinical development of a monoclonal antibody, anti-cancer, anti-rheumatoid arthritis, anti-cancer antibody, and their mechanism of the action. And for a concluded with the Indian scenario on the biosimilars. So, such a type of great personality is here for this session. So, on the behalf of this Department of the Chemistry, our college, Karmaveer Bhaurao Patil College, Vashi Autonomous, on the behalf of the principal, Dr. Suvankar sir, and the Shikshan Sanstha Satara, and on the behalf of all the participants of this two-day international conference, sir, we are very grateful for your sharing valuable knowledge. Thank you very much, sir.
Thank you very much. I appreciate the opportunity. I hope I was able to, create some interest into this particular subject, and hopefully, more and more people would start, getting involved in this particular, domain. Because as I said, this is a serious opportunity, and in a way, what Y2K did for the IT industry, I am convinced that COVID-19 is actually doing that for the biotech or the pharma industry. So, there is a serious amount of funding that is getting opened up for this particular, subject domain. And hopefully with more and more people actually creating this know-how, this knowledge, there would be some good quality research that can actually happen in this specific subject area as well. And I think chemists are very well, placed to come into this. I mean, it's not only a subject of biologists, but also chemists, because a lot of biochemistry or biological chemistry in a way is involved. Like I said, amino acids, these are basically chemical entities and very highly reactive chemical entities. So, we need people, chemists particularly, who can take up this particular subject with respect to both the properties of a molecule, biomolecule, as well as the analytics of this. So thank you very much for this opportunity, and all the best for the remainder of the sessions. I will have to take leave now because I have another meeting. Somebody is going to visit me. But all the best for the remainder sessions, and hopefully everyone ends up appreciating this particular effort of this two-day conference. Thank you very much.
Thank you, sir.
I'll take your leave now. Yeah, please, and feel free to share any questions with me in future. So Dr. Wadhwa does have my email address and everything. So Dr. Wadhwa, you can share it with the participants.
Yes, sir. Sure.
And in case anybody has any questions down the line, they can always share. Thank you so much.
Thank
bye
you,
bye.
Sir. Thank you, sir. Thank you. Lovely, sir.
Yes. Everyone?
Lovely, sir.
Hello.
Yes, ऐकू येते sir. Yes.
Welcome, Vedanath sir.
आवाज वाढवा sir. गवळी sir.
Yes, sir. I am Dr. Lakshman Gauri, Associate Professor in Organic Chemistry. It's my great honor and pleasure to introduce today's speaker, Dr. Viranna Empalli. Recently, he is working Assistant Professor, Punjab Engineering College, Chandigarh, India. Sir has completed his PhD in Organic Chemistry. Yes, sir. Hello.
सर गवळी सर आवाज येत नाही.
Hello.
आप कनेक्ट कर दो ना. शेयर कैसे करना?
Can I introduce?
Yes, sir.
I am Dr. Lakshman Gauri, Associate Professor in Department of Chemistry. It's my great honor and pleasure to introduce today's speaker, Dr. Viranna Empalli, who is presently working assistant professor in Punjab Engineering College, Chandigarh, India. Sir has completed his PhD in inorganic chemistry in 2012 from the University of Miami at US. He is expert in synthesis and characterization of air sensitizing catalyst precursors and multimetallic cluster complexes. Dr. Viran has worked as R&D junior engineer at AEME Telepower Limited, Haryana, in India during the year 2006-7. He has worked as a teaching assistant, as a working associate at University Miami, Florida, at USA during the year 2007 to 2012. Sir has worked as a postdoctoral researcher in Texas University at Qatar, Doha, during the year 2012 to 2016. Dr. Vedana has completed as a postdoctoral researcher at University of New Hampshire, Durham, at USA during the year 2016 to 18. Sir has worked as an assistant research scientist in Texas University at Qatar, Doha during the year 2018-20. Dr. Viranna sir has received many awards and honors with the Research Excellence Travel Grant in the 2014 and 2018 in Texas University. He has also received scholarship for the outstanding research from the School of Arts and Science, University of Miami at US. He has also honored with the scholarship from Indian Institute of Technology, Roke, in 2006. Dr. Viranna has published near about 17 research papers in reputed and international journals and also presented 12 research papers in international conference. With this brief introduction, I request Dr. Viranna sir, please start.
Thank you for nice introduction. Unfortunately, I am unable to connect to my webcam, but am I audible to everyone?
Yes, sir.
It's okay. I would try to start my presentation. So is it visible to everyone?
Yes, sir.
Everybody can see my content.
Yes, sir.
Okay. Thank you very much. I would take few minutes to thank Dr. Gurmeet Wadhwa for inviting me to give this lecture here as a part of this international conference. To be honest, this is my first lecture in India, I think. So I have returned from Qatar in January, and then all the things suddenly happened, like COVID and everything. And then it would have been better if we have had a facial or like a face-to-face conferences. But now, because of COVID, we have to live with this new kind of conferences, online conferences. Okay. So, without wasting much time, I would start my presentation. So, today I'm going to take you through the brief description about how the metal-based drugs has revolutionized the pharmaceutical industry. So, our previous speaker, Dr. Sameer Kulkarni, has nicely introduced the biologics of the drug development process. So I'm not going to spend much time on that side. He has made my work very easy. So I'm going to touch upon the chemistry side of the drug development, how as an inorganic chemist or like organometallic chemist can contribute to the drug development. So in this given 40 to 45 minutes, I'm going to try to expose you to the some of my individual researches that we have tried, and wherever it is appropriate, I would come up with some examples from the other researchers too. So, in a brief outline of my presentation, as I said, first few minutes I would spend time on what is the significance of the metals in the medicine, how the evolution of the metal-based drugs has started, and then I would slowly take you to the targeted therapy, which is you target to the proper tissues or the cells to have a maximum efficacy, like CO releasing molecules and photodynamic therapy. And then I would come up with some examples that we have worked on with the rhenium carbonyl complexes and how they can be used as a pro-drugs in anti-cancer treatment or the cancer treatment. And I will end up my talk with the future prospects and the opportunities what we have in this metal-based drugs field. Okay, so if you take metals, what is the significance of metals in medicine? We have plenty of examples from the day-to-day life. I am sure whoever is attending this presentation, they must have at least used once in their lifetime some of these salts we have, like ORS, oral supplements, and then this ENO, which is quite popular in India, ENO on, acidity gone. So we are quite familiar with this. So basically, these two simple examples that we see in our everyday life, they contain the metal salts like sodium and potassium in the oral supplements, electrolytes, and the magnesium and calcium carbonate salts are used in the synthesis or the preparation of these enos. So this is a quite simple example, and it's not that. Also, we can see the metals or the metal-based drugs have been used in quite a different variety of applications. You can see they are used in imaging probes and also in MRI scanning as a contrasting agent, and they are also used in treatment of several diseases like anti-cancer and rheumatoid arthritis. And we also see the application of metals in the dental implants and then in the other knee replacements and all those things. So we know that the metals are everywhere in the medicine, and we are going to see how this drug development process or the use of metals in the medicine has been evolved through the time. So let's go to the first slide. We are all familiar, especially we Indians are fond of gold and then also silver and then the copper. So these coinage metals have been used by our ancient civilizations from several centuries for different purposes. Some of them might have used for ornaments, some of them might have used for preparing warfare agents, but also there have been some indications that the gold and then the copper has been used by the Egyptian civilizations and also the Chinese in some medicinal applications. And presently, if you see the gold based gold metal based drugs or gold therapy, we can say, has been used in different disease treatments like rheumatoid arthritis that I have said, alcoholism treatment, depression, epilepsy, and migraine. And also the copper, we Indian civilization, we have this practice of throwing the copper coins into the rivers. So our ancestors, by knowingly or unknowingly, put it into our tradition to throw the copper coins whenever you visit the holy places, and you throw your copper coins into the water. So the obvious idea behind that is that maybe our ancestors must have known the antimicrobial properties of the copper, that they treat the water, they sterilize the water. So maybe that is the reason why our ancestors prompted us to throw the copper coins in the water. Along with that, we have also mention of the use of the copper in our Vedas with the name Tamra. The Tamra stands for the copper, and the different formulations of the copper powder has been used to treat the various diseases related to the breathing and then the digestion properties and all those things. So definitely the metals, especially the coinage metals that we have been seen in the history, like gold and copper and silver, they have been also used by our ancestors and various civilizations in the treatment of the medicine. And the silver, as you see here, silver also has been known for their antimicrobial properties. And then silver has been used in treating the burns. The medicine silverine has been developed to treat the burns treatment. And also it has shown properties to treat brain infections and the skin ulcers. So definitely there are a lot of wide variety of applications of the metals in the field of the medicine, as we've seen from the data and as we've seen from the history perspective. And if you take a human body, what is the significance of metals in the human body? So the metals plays a crucial role in the form of metalloenzymes, or as a cofactors. They catalyze key important reactions which are crucial for functioning, physiological functioning in the body. Here are the two examples, as you can see here. These are the one of the hydrogenase enzyme, which contains the active center of iron and nickel. And there are two models of these hydrogenase enzyme in which we have two metals, iron and nickel. In another one, we have two metals of the same category, the iron and iron. So these hydrogenase enzymes, as I've said, they kind of catalyze this simple reaction, oxidation of hydrogen. And here you can see that hydrogen is converted into protons, and this is a reversible reaction. Depending on what type of environment is there around the metals, the direction of the reaction changes. So suppose that if you have electron acceptors around the metal center in the active site, then the hydrogen gets oxidized to produce the protons. And if you have electron donors in the vicinity of the metal center, you will have production of the hydrogen molecule, which is the reverse of what we have shown here. So this is a simple and classical example of the importance of the metals and their geometry in the different biological molecules like metalloenzymes we have seen here. So depending on the environment around the metal and the electronic properties around the metal, they try to activate different parts of the biological molecules, and they activate and they participate in different reactions. And also here you can see another example, which is a another metal-based enzyme, which we call copper zinc superoxide dismutase. So importance of these enzymes in our human body is they create or they prepare us, our body, against the superoxide or the free radicals that kind of inhibit our cell death. So these reaction here, you can see the free radicals are converted into the free oxygen and hydrogen peroxide using this molecule, which we have been shown here. So here you can see that there is an interaction between the two metals. They are communicating through a linkage here. So here the zinc metal has a role in the activity of the enzyme to bind to the proteins, and the copper is involved in catalyzing this reaction. So here you have a mixture of two metals where they are communicating with each other, and each metal has its specific function. So the nature is teaching us something to learn from the biological molecules, that two is better than one. So sometimes if one compound may not be active for certain properties, but if you add another material, another metal into that, it might act as a better drug or a better function that you are desiring to. And also we have these iron, zinc, and copper metals, which have been classified as essential trace metals. Their presence is crucial in our body for definite functions. For example, iron, if you have a deficiency of iron, you would face a disease, anemia. And if you have a deficiency of zinc, you would have again a problem with the growth and also the loss of sense, smell, and taste that we are facing with the COVID-19 symptoms. So these are the importance of the metals in our human body. So without much going into the basics, I'm going to start with some of the triggering points that has triggered the revolutionization or the catalyzation of the field of metal-based drugs. So if you take a simple example here, an important contribution by Dr. Paul Ulrich, we can call him as a pioneer of chemotherapy that we are using for treatment of the cancer. So he has developed an arsenic-based metallic drug, which is known as magic bullet. So everybody must have known what does it means by the magic bullet. So you are targeting a bullet. If you want to shoot something, you want that bullet to be going to hit the target right in the center, in the middle of the point where you want to point it. So the Paul Ehrlich had this idea that whenever we are using any drug or any chemical, it should specifically treat for that particular disease that you are targeting. So he has developed this Salvarzone 606 medicine, especially for treatment of syphilis. So he can be called as the father of the chemotherapy, because at that point of time where it's rarely a mention of how the disease is going to interact with the chemicals or metal-based drugs, which is not known clearly at that point of time. He kind of tried 606 different varieties of the medical drugs. He has tried them. And the reason why it is called 606 is because the 606 compound he has tried has worked with the better results for the treatment of the syphilis. And he has also a strange, an interesting story behind the development of this magic bullet. So this arsenophenamine drug they have developed when they first experimented, because of the experimental errors by his student, it was neglected, and they tried to neglect it, and they went for the 607 compound. But when a Japanese student joined Dr. Elrich, he reinvestigated the experimental results of the compound 606, which is assay phenomenon, and he said that there is an error in reporting the data, and he redone all the experiments, and then he came up with the results, and he showed the results to the Paul Elrich, and they were quite happy, and they were successful in developing this metal-based drug. So we can say that this is one of the interesting story or interesting step where a metal-based drug has been used for the targeted therapy. And similarly, there is another drug developed by Robert Koch. So Robert Koch was doing research on using the gold in the different treatments. So he was interested in certain gold salts for treatment of tuberculosis. So he found that gold cyanide or the gold salts were very effective in killing the bacteria tuberculosis. So he has been formulated different gold metal based drugs, and you can see here one of the gold metal based drug which has been developed and commercialized for various applications. So arenophin is being used for the treatment of rheumatoid arthritis, and this is also been used recently. Arenophin has been repurposed to treat various diseases like AIDS, cancer, and recently some researchers are also trying to repurpose this existing drug, arenophin, to treat the COVID-19 symptoms. So we can see that the metals were different metals starting from gold, silver, and arsenic. Different metals were used by during the 17th, 18th, and 19th century to treat various diseases. And they were well known that the metals plays a crucial role in treatment of certain diseases. And by the end of the 19th century, there was more interest in relating the activity of metal-based drugs to their structures and the ligand environment around the metal, and what is the role of the metal in imparting that particular property to the metal-based drugs. So as we proceed to the 20th century, we have an accidental discovery. So we call this an accidental discovery of cisplatin, which is a chemotherapeutic drug which is widely used in treatment of cancer now. So it has been developed by Rosenberg at Michigan State University 50 years ago. It's a simple inorganic salt. You can see the structure of this metal-based drug. Here the metal is platinum. Platinum is in a square planar structure. It is platinum ammonium chloride salt. It's a simple inorganic chemical salt which has revolutionized the new era of bioinorganic chemistry. So after the successful use of this cisplatin for the chemotherapy, several inorganic chemists has tried similar platinum-based drugs which has a square planar geometry, and similar gold complexes, which has similar square planar geometry, and palladium complexes with a square planar geometry, and they have tested their anti-proliferative activity. And this is definitely a triggering point for the new era of bioinorganic chemistry, and the metal-based drugs research has been catalyzed by the invention of cisplatin. And again, whenever we talk about the metal-based drugs, these are the key factors which has to be keep in mind: the geometry, the mode of action, and then the localization. These three are the important driving forces which kind of decide what is the use of a particular metal based drug in different applications. So as we know from the nature, different enzymes have active sites, and they have lock on key model properties where a particular metal goes and binds to a particular ligand in a specific geometry only. So the geometrical confinement is the key. So we have to, whenever we are designing a metal based drug, as Dr. Samir Kulkarni mentioned about the biologics, when you decide the target, you have to know what you are targeting. Similarly, whenever you are developing a metal based drug, you have to keep in mind what is your target, what is the receptor molecule you are trying to bind your metal to, and you need to know what geometry is available there and what kind of coordination complexes you have to prepare. And then the mode of action, which is kind of another important property that a metal based drugs has to be considered, like whether it is going to act as a toxic drug by chemotherapy, or it's going to act as a prodrug, and what is its role, how it is going to act on that particular target. You need to know that in detail. And also the localization, or we call the biodistribution, how the drug or the metal based drugs are being localized, they are uptaken by the cells, how they are taken by the cells, whether they are soluble in aqueous environment, because our body has water. So the metal, many of the inorganic chemicals that are being developed as a metal based drugs, they have this deficiency because they are soluble in non-aqueous solvents. So this is another area the metal-based drugs development has to concentrate on. So with the advancement of novel bioanalytical techniques and novel rational design of ligands, this new era of metal-based drugs has been brought into. And today I'm going to give some examples from our research group, which I have done some research on some metal complexes, and how I have tried to visualize these metal complexes in different applications related to the medicinal field. So here, this is a classic example, a simple platinum tin complex that I have prepared as a graduate student when I was in my first year of my PhD. So you can see here how the metal complexes have these unique features that I have stated here with this example. So here in this complex, platinum has two tin groups and two isocyanate groups, and it is a distorted seesaw structure. And you can see that the geometry is dependent on the substituents on the tin and as well as the substituents on this ligand. And here the angle between the two isocyanates is 159, and the oxidation state of the metal here is platinum is in +2 oxidation state. And when you expose this molecule to the small molecules like hydrogen at room temperature, the platinum converts into platinum 4 oxidation state. And you can see that the geometry has been changed here depending on the change in the oxidation states. So this is the unique features of the metals that are not available in organic based molecules. So somebody might ask why we want to go to the metal-based drugs when there are several biomolecules, several organic molecules are there already developed for certain treatments. Why people are interested in metal-based drugs? Because of these unique features. The coordination geometry, as I have mentioned, it changes with the change in the metal environment and then the electronic environment, as you see in this example. And then the metals can also act as electron transfer agents, and they can transfer the electrons whenever it is necessary. And then they can also act as the reservoirs. And another important property the metals have, which the other organic molecules also have, is the binding affinity with the proteins and other molecules of interest. So when I go forward into my presentation, you can see how this binding affinity plays a crucial role in developing the metal-based drugs. And also we have a certain facility with the metal-based drugs. We can probe how these metals are interacting with the different organisms inside the human body. Okay, so when we talk about the drug development, currently we have a lot of opportunities with the metal-based drugs, and we have come upon new approaches. Like we can see that I have already mentioned about this repurposing the existing drugs. So the COVID-19 has thrown as a challenge to develop the vaccine against COVID-19 within a short time. So in this short time, if one has to start from basic novel design of the drug and try to take it to the market, it would take surely for a longer time scale. So what the scientists are doing now is trying to use existing drugs and repurpose them to treat the COVID-19. So this is an important area or opportunity where several metal-based complexes which have shown some properties like antiviral properties or antimicrobial properties, they can be used for treatment of COVID-19. They can be repurposed. Okay, and again, to the time frame involved in screening different metal complexes or metal-based drugs. Like let's say you have designed a ligand, you complex it with metal, and you want to test its activity, medicinal activity or anti-proliferative activity, and you made 10 or 15 different ligands, and you want to try how the ligands are playing a crucial role in imparting that particular activity. So it will take huge amount of time if you test one drug at a time. So the high throughput screening is an important facilitation where you can test n number of drugs. Hello? N number of drugs at one time, which saves your time. And also the structure activity relation, which is being mentioned by Dr. Samir Kulkarni again, it is also another key development where whenever we are designing a ligand or we are designing a metal-based drug, we need to understand how the ligand is playing a role and how the metal center is going to play an important role. The electronic properties of the ligand and the electronic properties of the metal and how they are going to communicate with each other and how they behave with the organisms, that has to be known. So the structure activity relationship is an important area where the 21st century scientists, inorganic chemists, biologists, they are playing an important contribution in this field. And again, an important area in the drug development is the targeted drug delivery. Again, our previous speaker has touched upon this topic, how monoclinical antibodies can target a particular tumor cell with the specificity. And this is an important area where the metal complexes has been used. I'm going to give some examples in this particular topic of targeted drug delivery. And again, the metal complexes synthesis involves a lot of toxic metals sometimes heavy metals. And one has to think about the synthetic methodologies we are adopting, whether is it safe for environment. So we are in era of where we have done huge damage to our environment. So we don't want to treat human diseases at the cost of the environment. So when we design metal-based drugs, one has to think about the synthetic strategies we are going to use, how effective it is. At the same time, what method of synthesis you are choosing, what kind of solvents you are choosing, what kind of ligand structures you are choosing, whether it is hazardous, whether it is okay to use in environmental conditions, that one has to treat. And again, a new era with the advent of computational simulations, this can reduce our effort in the laboratory, especially all those chemists who does organic synthesis. They know much about the tedious process involved in multi-step synthesis. Like a simple drug can involve more than 20 steps of synthesis. And by the end of 21st step, if you think that your compound is not right compound for certain activity, and that amount of time you invested in synthesizing that particular compound is complete waste. So now most of the companies, they are trying to use computational simulations before even starting their synthesis in the laboratory scale. So they are going to try identify some candidates, and they are going to run a simulation to see if that particular combination of metal and the ligand environment is ideal for treatment of that particular disease. And they are going to shortlist 10 to 15 candidates, and then they are going to give those projects to their R&D scientists, and they will work on those. So when we talk about the key challenges involved in metal-based drug development, these are the key challenges. These are the important steps in the drug development. And at each step, when you say target, dosage, transportation, efficacy, and excretion. So one of the key challenge that we have in the metal-based drug development, as I have mentioned before, it is how we are going to control the dosage of the metal-based drugs. Some of the metals, they are very effective, and they have shown excellent in vitro properties, but for in vivo properties or in vivo analysis, they have not been suitable. The main reason why they are not suitable because they have a poor solubility in water. So when you develop a metal-based drug, this is an important challenge you have, is that if your metal-based drug has only solubility in non-aqueous solvents and it is not soluble in water, then no matter how efficient it is in vitro analysis, when it goes for animal studies or when it goes to the later stage of the development, then it is going to face a problem, because if it is not soluble in water, the dose of the metal available for treatment of that particular ailment is in very low quantity because of its solubility. So certainly the solubility is an important challenge for the metal-based drugs. And also another important challenge for the metal-based drugs is the excretion. So you are injecting a metal-based drug into the human body, and when you are injecting it, you also have to think about the strategy where it is going to end up after it has done its purpose. So suppose you have used a cis therapeutic agent, the cisplatin, and you have injected into a patient for chemotherapy. And it has been known that whenever a patient goes for the repeated chemotherapy sessions, so when a patient goes for the repeated chemotherapy, he has a problem of side effects, like different side effects of nephrotoxicity and a lot of other side effects. So if we don't have this exit strategy like we have in all the business plans, we need to have a strategy for even for the metal-based drug development also, where from which channel this metal is going to exit out of the body. Whether it is safe to stay in the body for longer time, we also have to think about that. And if it is not exiting from the body, what are the other strategies we can use? Whether we can bind it through some organic molecules, which makes its excretion easily, and all those properties. And these are the important challenges. And also there is a challenge of the transportation. So again, if you want your drug to be effective, it has to reach the target in a right manner and in a proper dosage. If it fails to reach its target, and if it is not depositing in a proper cells where it is intended to, then again we will have a problem. So we need a proper mechanism and a proper strategy to transport the metal-based drugs to the desired target. So for this purpose, currently there are several options like nano carriers, nanomaterial composites. Again, I am going to give one example of how we can transport our material to the desired target. Okay. So continuing with the key challenges we have, as we see that even after 50 years of the development use of cisplatin in chemotherapy, which is a simple platinum compound, inorganic solvent, inorganic compound, we are still faced with the problems that there is no inorganic metal based compound exists in a approved stage. So still there are ruthenium based, iron based drugs which are there for the treatment of the cancer, which are in the clinical trial stage, but none of them has been went out of the clinical trials, and none of been them have got approval. So can you imagine, even after 50 years, with the first cisplatin complex known for the chemotherapy, we are still unable to develop a metal based drug for the key disease, which is the cancer. Okay, so cancer is still considered as an incurable disease. So we have a lot of opportunities in this area where a different strategy can be adopted to improve the efficacy of metal based drugs, to make them effective against the cancer, and to take them beyond the clinical trial stage. And it is surprising to see that when I have searched how much time and how much cost it involves in developing the drugs. So according to a data, approximately starting from the registration of your drug, by the time you get an approval from the FDA, it involves approximately 10 to 17 years. So can you imagine that many years, 10 to 17 years of time involved from the day you registered your drug for certain disease and the day that drug got approved. And how much cost it involves? Almost $1.5 billion. Our previous speaker, Samir Kulkarni, has done the calculations how we can convert billions dollars into the Indian money. You can imagine $1.5 billion is involved in getting the approval. So definitely there is a big challenge here in the time and cost management in developing the drugs and taking them to the market and available for the use of the public. And as you see here in this slide, there are still several diseases which are uncurable. Dengue fever, we know there is a medicine for the dengue fever to minimize its side effects, but accurate drug to treat the dengue fever is still unavailable. Similarly, we know AIDS is uncurable. We can cure the symptoms, but still it is there from several years. And also the recent challenge we are facing, all the humankind on the globe, we are facing the challenge of the COVID-19 virus. So several scientists are trying to race up with the time to come up with a vaccine, but still it's in the trial stage, and we hope that the vaccine gets successful and we have a vaccine by end of September, as Oxford University is promising. So again, the drug resistance is also a key challenge. So one of the ideal example for the drug resistance is the cisplatin. I have already given why the drug resistance plays a key role. So if a cancer patient is treated with the chemotherapy by administering the cisplatin, and if he has to go to the repeated chemotherapy, and then that particular drug develops the resistance, so that particular cancer cells, they get the resistance for the cisplatin, and even if you uh, give that particular drug again and again, then it doesn't give you the intended effects as you treat. So this drug resistance has to be kept in mind when you design. So design a metal based drugs, and again the gene mutations, which is also a key challenge for the drug development. So like we are reading in the news that the COVID-19 is changing its gene structure from time to time. So when it started in November or December as the Chinese state, if it is still in the same form or it has changed into its different variations, we don't know. So that is another challenge that we have when we try to target the drug development. So these are the important aspects one has to consider when we go for the drug development. And here I have found an interesting data regarding the COVID-19. So in one of the US hospital, they recorded a number of patients who are admitted into the hospital. So they were surprised that when they compared the data of the patients admitted into the hospital, there were more number of non-smokers who have been admitted into the hospital because of COVID-19 compared to the smokers. So this is a good news for the smokers, but don't jump in a joy right now. It's not yet confirmed. So they were surprised to see that the number of patients who are non-smokers are more chances of admitting into the hospital compared to the smokers. So then deducted what is the reason behind why the smokers have this God gifted resistance, you can say, is because when you have a smoke, it induces the development of hydrooxygenase enzyme, which has been used in a treatment of, which has been shown that it upregulates the antiviral properties. So that is the reason why the smokers have kind of advantage over the non-smokers during this COVID-19 times. And again, there is a huge opportunity with this known traditional drug. We always use turmeric powder in our Indian culinary. In all majority of the Indian cuisine has a turmeric as a major ingredient. So the turmeric powder has this ingredient curcumin, which has been shown to have important antiviral properties, and it has also been shown to have anti-proliferative properties, and also it has been used by some researchers in Kerala. They have treated the nanomaterials with this curcumin, and they tested its efficacy in treating the COVID-19. So again, there is a lot of opportunity for using this curcumin as a ligand. As you can see here, this C double bond here and this keto-enol place here is a ideal place to bind the metals, and one can use this metal curcumin complex and try to see whether it is effective in all these incurable diseases that we have listed here. So these are the key challenges, and these are the history of metal-based drug development. So now I am going to give you a new field which is in hot topic now, which is targeted therapy. So as I have said several times, my previous speaker has already mentioned what is the targeted therapy. So I'm not going to spend more time on this field, but I would try to briefly introduce you to the concept of photodynamic therapy, how photodynamic therapy based on the metals kind of can be used for treatment of cancer again. And also this topic is also being mentioned by our previous speaker, antibody drug conjugates, how antibodies can be conjugated to the drugs to improve their efficacy again. And this is also an important area where metal-based drugs play a key role, especially the metal carbonyls. Metal carbonyls like manganese and rhenium, they have been extensively used as the CO releasing molecules. So I'm going to explain these two fields, photodynamic therapy and CO releasing molecules, with some of the examples we have done in our lab. And also there is another mode of approach that they say that we need to bind a target molecule, certain molecules, which are going to have a huge accumulation in unhealthy cells. So this is a preferential accumulation of some molecule in unhealthy cells. So we need to identify those kind of molecules and try to tag them to our metals, and then we can have an improved efficacy of metal-based drugs for treatment of that particular disease, whether it be cancer, whether it be arthritis or anything. So this topic is also being touched upon by our previous speaker, so I'm not going to spend much time on that. And another important emerging area is the pH responsive drugs. So you design your ligands in such a way around your metal center in such a way that in a psychological pH conditions you have, it responds in a different way, triggering some signals or triggering some definitive cell death mechanisms. So this is another important area where a targeted therapy is designed. Okay, so I'm going to spend few minutes on this area, CO releasing molecules. So I'm sure many of you, the chemists who are working in inorganic chemistry, you must have come across this topic. Okay, the CO releasing molecules, why they are important? Because they have it has been found that the CO as a therapeutic agent, it has important properties like a vasodilation, anti-inflammatory, antimicrobial properties, reperfusion injury, and all these kind of medicinal therapeutic advantages. So how we are going to introduce CO into the body? Okay, so traditionally when they use the CO for therapeutic purposes, they introduce it through our lungs. Okay, so they mix it with air and they introduce through the lungs. So this is one of the clinical way of introducing the carbon monoxide into human body. Okay, but what is the role of metal based drugs in this field? So this is an alternative way of introducing the CO into the human body for desired purpose, as it is shown here. So what we do is we take metal carbonyl complexes, so we dissolve them in appropriate solvent and we make a solution and we inject into the desired place. So these metal carbonyl complexes are inactive in the body until they are triggered. So the trigger can be either a light of suitable wavelength or a thermal trigger. Then it releases the CO, and those CO molecules can be absorbed by the cells or tissues, and then they can be used for these various kind of medicinal purposes. So these CO releasing molecules are mainly based on the metals, and there are also several examples exist with non-metals or the organic molecules which are also CO releasing molecules. But what is the advantage of metal based CO releasing molecules? Because we have this opportunity to track their distribution in the body. So we have these carbonyl tags, so we can monitor their distribution through the IR spectroscopy, and we are releasing the carbonyl molecules. The metal carbonyls are acting as a reservoirs. So whenever the body requests the CO, they release the CO by the trigger. So not like the organic molecules. Here we have a control. So we have a control on when the CO molecule will be released, and we have a control and where it would be released. So that is the advantage of a metal-based drugs compared to the organic molecules in the field of CO release. So this is the first compound I have prepared in Texas A&M University at Qatar. It's a simple compound. We take metal carbonyl, manganese pentacarbonyl, and we use this ligand, and a simple reflux gives this beautiful compound. So I have prepared this complex for other purpose, for the CO2 reduction, because we know that these kind of ligands are helpful in the CO2 reduction. But when I try to do some chemical analysis, like when I want to take its IR in exposed to the visible light, the compound when it is soluble, when it is dissolved in any solvent, it is blue in color, and by the time I take it to the IR spectrometer, it turns into yellow color. So I was surprised to see this color change. What's happening? Is my compound decomposing, and why this color change? So I tried to monitor it through the FTIR, as you can see here, the FTIR spectrum. So the manganese carbonyl complex has 3 COs, and all the 3 COs intensities are decreasing when we expose this carbonyl complex to the light. As you can see, 0 minutes, 20 minutes, and by 35 minutes, all the 3 CO molecules has been liberated from the molecule. And also you can see a free CO here. So this is an advantage of using the metal carbonyl complexes, because you have this CO which acts as a spectroscopic tool. You can trace how much of the CO is released, and you can also trace whether one molecule of CO is released or whether all the COs are released. And then you can also study what is the fate of your metal complex after it releases the CO. That is an important challenge in the CO releasing molecules field. So there are many metals which release the CO, but what happens after it releases the CO, whether it is stable or whether it is toxic to the human body, those kind of studies are need to be done in much more elaborate way. And another interesting factor that has here is we are irradiating this complex using the light. So this is the absorption spectra of this compound here, and it is having a huge absorption band here around 560. So we take this compound, we irradiate using the 562 nanometer radiation light laser, and then we keep it in the dark, and it releases the CO, and we track the CO release using the FTIR. So this is an example how a metal inorganic chemist or organic chemist can try to use the chemistry in an effective way. Okay, so this is another example of the CO release from our own lab. So this is a rhenium carbonyl complex with a pyrrolidine ligand here, and this compound, when you heat it in a solution in the presence of any ligands, it kind of liberates one molecule of CO, and then that ligand binds to the metal. So this is another way of preparing the CO releasing molecules. Here we are triggering the CO release by heating. The previous example was CO release using the light. Here the CO release is using the heat. So when you heat this compound in a solution of any solution with the presence of any of these ligands, it liberates the CO. So again, we have followed how a tetracarbonyl complex, here you can see the IR spectra, is going into the tricarbonyl complex, and we followed its reaction progress through the FTIR. And I'm going to show you a short video. And this is again, we use the same molecule for a different purpose. So we use this molecule as a catalyst for hydrogen release from the ammonia borane. So if you have time, we can see this video. So this is a short video. So here I take my rhenium compound, I dissolve it in toluene, I add ammonium borane to that, and you can see that at room temperature, it starts releasing the hydrogen molecules. So this is a reactive compound. It liberates the CO, and it also provides an opportunity to bind this amine to the metal, and then it liberates the hydrogen. So this is an important paper that we published in organometallics. So the same molecule we have used for CO releasing molecules, and we have same molecule we have used for as a catalyst for hydrogen release. So this is the beauty of the metal complexes. So we prepare the metal complexes for certain applications, but there is always a chance of using the same metal for a catalyst for different reactions. And we can also change the ligand structure, and we can modify the ligand, and we can repurpose the same material for different applications. So this is an example I wanted to show. And this is another CO releasing molecules we have prepared in our lab. So the molecular structure, crystal structure is shown here. Again, it is a rhenium based metal complex. It has 4 carbonyls. Again, when you dissolve this compound in any of the solvent at room temperature, it liberates the CO molecule. And it has a capability to bind to any molecule because this hydrogen is acidic. We can conjugate this place to any organic molecule for intended purpose. And also, you can see here how, again, we have used FTIR spectroscopy to monitor how this carbonyl complex changing from tetracarbonyl to tricarbonyl, and this process completes within 24 minutes at room temperature without heating. So this is an example of room temperature CO release from the organometallic complex, which is having a rhenium and a ligand, which is capable of binding to the molecule of interest. So there are various ways in which an organometallic chemist or inorganometallic chemist can contribute to the development of metal-based drugs for the different purposes. So as I have said, again, the intended purpose of using this complex was different. we try to make complexes for CO2 reduction, but we observed that when we dissolve this metal carbonyl complex in solution, we saw change in the IR spectra, and we have repurposed the same molecule to study its CO releasing properties also. So similarly, this molecule also has affinity for amine molecules. So we are now in a process of testing this compound to see if you can take this molecule and try DNA binding studies. So, and this molecule also has an interesting property that it activates the triple bonds and it gets converted into a double bond. So just I want to ask to Gurmeet, how much time I have, sir?
So 5 to 10 minutes more. 5 minutes.
Okay, 5 minutes more. Okay, then I would try to go quickly to finish it in a time.
Yes,
okay, so as I've said, this is an important area, the photodynamic therapy, which is being used for several years. And this is a brief pictorial representation how the photodynamic therapy works. So you have an inactive drug, you inject it into the human body, and you kind of wait till this drug populates all of the cells, and then you trigger it with the light of appropriate wavelength, and then the pro drug or inactive drug becomes active, and it starts acting on the desired target by killing the tumor cells or anything. So here the metal carbonyl complexes has been used in this area, and one of the key challenges the metal carbonyl complexes that are used in this application have is their absorption is in the UV visible range. And for ideal application in photodynamic therapy, one has to have absorption in the near infrared range or the red light range, because when you irradiate your skin with the UV light, it doesn't go deep into the tissue. So it has to be going deep into the tissues, then it has to be near infrared light. So this is again an example from the marker who has used a rhenium-based metal complexes with the carbonyls, and they have used this to target the cancer cells. And they have observed that when they use a light of 365 nanometers and irradiate this complex, and it's killing the tumor cells through 3 different mechanisms. In one way, it is interacting with the oxygen present in the tumor cells and generating a reactive oxygen species that kills the tumor cells. And it is also releasing the CO molecule, as we've seen that the CO molecule also has anti-proliferative properties. This is also contributing to the killing of the tumor cells, as well as the metal carbonyl complex itself has intrinsic cytotoxic properties that also contribute to the death of the tumor cells. So this is a classic, beautiful example of how the metal carbonyl complexes has been used in the photodynamic therapy. And this is again similar to the photodynamic therapy, but we call it as a photoactivated chemotherapy. So the difference between the photodynamic therapy and photoactivated chemotherapy is just the mechanism of the action. So PDT only works when you have oxygen-rich environment, because it has to interact with the triplet oxygen to generate the reactive oxygen species. Whereas in the PACT, you don't need that restriction. You can simply take your metal carbonyl complex or the product, and then it acts because of its cytotoxic activity, and it works even in the oxygen deficient cells also. Okay, so this is another important area we are been trying to step our foot in, which is the multi-modular approach. As you can see here, we can use the same rhenium metal carbonyl complexes, and this rhenium metal carbonyl complex has the CO tags, which can be used as an imaging tool to monitor how these metal complexes distribute in the body with a different kind of imaging techniques. And this carbonyl complex has a functional moiety which can be conjugated to the molecule of interest. The molecule of interest can be antibodies, it can be any receptor molecules, any proteins, which preferentially take this drug to the desired target. And we have a CO molecule which is labile. It can be freely released and it can bind to the DNA. So we are trying to explore this metal carbonyl complexes in and its cytotoxic activity. And we are intended to develop these kind of nanocomposites where we have a nanomaterial loaded with the metal carbonyl complex, and it's coupled to the molecule of interest, and we can have this several advantages of cytotoxic studies, live imaging of the cells, and then we also can understand the mechanism of action. So I would try to go to skip few slides. So these are the results we got from the compound we have tested, and we have tried to understand how the secondary coordination sphere affects the cytotoxic activity, and how the ligands affect the cytotoxic activity. So these are the results which are being performed by Hamad Medical Corporation in Qatar. We see that when we have a methyl electron donating substituent, cytotoxic activity is more, and when you have electron withdrawing substituent, cytotoxicity is less. So this is again the metal carbonyl complexes or the metal based drugs offer a different area where we can explore their cytotoxic activity as a anti-cancer drugs. So this is another compound we have tested in various cells as is shown here, and this is also showing an important anti-proliferative activity, and these are the results. So it has IC50 value of 6.24, and we are currently investigating how we can proceed further using these metal carbonyl complexes. So this is an important Dr. Gurmeet, can you give me 2 more minutes? I will wind up with this important example. So here we can see this vehicle. This is an example of a courier boy trying to deliver his courier. So we can imagine our nanomaterials similarly as a drug delivery vehicle. So we have our metal complexes, which are drugs, which are the load, we can say, and we have a carrier, which is the nanomaterial. And as you can see here in this picture, these are the nanomaterials we are trying to use, and these are the materials we are trying to use as probes. And we load them on the nanomaterial, and we tag them with the antibodies, which can take them to the desired target, and then we try to study their purpose. So here, these are the questions we want to ask: whether it reached to the proper place, whether it is safe to use, and all these questions we want to ask ourselves when we use the nanomaterials as the carriers. And these are the important points one has to consider when we use the nanomaterial based carriers, how these factors are playing an important role, like biodistribution, bioanalytical techniques like different imaging techniques, confocal microscopy, ICP-MS, and all those things. So I want to conclude my talk by saying that we have several opportunities with the metal-based drugs. We can optimize the structures of the ligands in order to address the key challenges. We can come with the novel combination therapies. So if chemotherapy is not working, let's try to hook up the chemotherapy with the immunotherapy Dr. Samir Kulkarni has mentioned. And we can also concentrate on repurposing the existing drugs for the different purposes. And also there is a huge need for interdisciplinary research. So chemists cannot do it alone. We need biologists, biochemists, and biotechnology people to properly design a metal-based drug which can be having a huge impact on the incurable diseases. And also there are like several opportunities exist with the multimetallic cluster complexes for different purposes and for theranostic applications. So with this, I want to thank everybody. Thank you for your attention. And I want to thank all my collaborators in Hamad Medical Corporation and my postdoc advisor for giving me an opportunity to work on this project. Thank you all. Thank you very much for giving me this opportunity. So I would say thank you. This is the end of my talk. Okay.
Thank you, sir. Lovely, sir.
Hello.
Yes, sir. Lovely, sir.
Sir, one question is there.
Yes.
Is it possible in human body to reduce the sodium metal concentration by replacing any other metals like potassium?
Uh, I am sorry to say that I am not that much knowledgeable in how these metals regulate different electrical properties in the body. By profession, I am an inorganic chemist, so I know much about metal carbonyl complexes and how they behave when we treat with the different conditions. So I am unable to answer that question. Sorry.
Another question.
Uh,
Now the Ayush Mantralaya, that arsenic
सर
tablets
आवाज येत नाही.
for the COVID-19.
mhm.
So arsenic is a heavy metal, so it is poisonous or not to human body?
Yeah, there is a big discussion about whether we can use heavy metals in medicinal purposes or not. So there is still a lot of contradictory statements. Some people say no, we should never use any heavy metal based drugs like a mercury, arsenic, or antimony. But some people say no, if they are showing intended purpose in a minimal dosage, then we can use it. So I would say if it is in a minimal concentration that body can tolerate, and as I stressed upon, if we know the excretion strategy, that if we know for sure that it is not going to stay in the human body for longer times and there is a proper mechanism for its excretion, then I would say it is okay to use as long as it meets the purpose. Again, we have to strike a balance between its efficacy and its efficiency as well as its dosage. Since it is an heavy metal, we need to go with the minimum dosage and also to see that it's properly excreted from the human body. And if you are not sure that it's going to be excreted, then I would say don't go for it. So that is my perspective, but different people may have different perspective regarding this topic. And as it is open for discussion, like many people have many opinions, so that's my opinion.
That was sir.
Sir, proceed.
Okay.
Hello.
Hello.
Gowri sir, vote of thanks.
Yes, yes. Now I am here
Vashi,
to
Vashi.
propose the vote of thanks. A big thanks to Dr. Vedanna Empalli for his efforts towards these metal-based drugs. I must mention our deep sense of the appreciation for the Dr. Vedanna for his detailed explanation about this importance of metals in medicine, unique features of metals like changes in geometries, importance of metals as antimicrobial activities like the selenium, copper, gold, arsenic, etc. And he has discussed about this importance of the challenges and opportunities. So all the participants from this lecture must be motivated to design the new drugs and may be useful in the society. On behalf of Department of Chemistry, Rayat Shikshan Sanstha, Karmaveer, Bhaurao, Patil College, Vashi, and organizing committee, I thank to Dr. Vedanna sir. Thank you very much.
I thank all the organizers for providing me this opportunity and all the participants. Thank you very much. Thank you.
Yes, I'm here. Good afternoon. Thank you, sir. Thank you very much for kind introduction. Am I audible? Yeah, thank you. So thank you, sir. I think first of all I should thank the organizers of this international e-conference. I think the situation is all everyone is aware now. We are seeing this, through media, paper, but I feel this is a good opportunity that teachers are trying to engage students and and other faculty also to such e-conferences. I feel it's really a good idea, and I congratulate Karmaveer Bhaurao Patil College for organizing such event, and especially I'm thankful to Principal Sir Dr. Shivankar for providing me this opportunity to present, and special thank to Dr. Vadva for requesting for arranging all these talks. It is an interesting session. So, I should also thank Dr. Samir and Dr. Virana for nicely creating a background for my talk, because we in last couple of hours listened about biologics, and how they are into drug development. Dr. Virana has nicely elaborated on drug discovery approaches, targeted therapies, and he has also given what are the recent research going on in the lab, and how it is related to how he's working at the interface of chemistry and biology. With this background, I think today what I wanted to do, I wanted to tell what is the role of synthetic chemists in the development of vaccines. So, as a chemist, or as a chemistry graduate, master student, or a PhD student, we know that we are some or other way trying to contribute to a drug discovery process. We try to develop reactions, try to develop processes, then we publish it. Directly, we try to work in the field of medicinal chemistry and try to develop bioactive molecules, or sometime our work, some other medicinal chemists or biologists, they refer to our work, and then they use that for in the field of medicine. Hello. So in this event, I feel that as a chemist, we are trying to directly or indirectly trying to contribute in the field of drug discovery. But now, in last 4 to 5 months, we are all hearing so much about vaccines. And this particular talk, I want to highlight how chemists, what is the role of chemists in this field? How can we contribute towards the development of vaccines? I'm unable to. Hello, sir. Hello. Yes, sir. My slides were they were hang. What I wanted to tell that the situation is bad. COVID-19 is already announced a pandemic by WHO, World Health Organization, and is a current global health crisis. According to WHO, now you know that in less than 4 to 5 months, COVID-19 has spread through almost all countries and regions. You can see here the data what I collected on May 24th. You can see there are already 53 million confirmed cases of COVID-19, and India is also listed here. You can see around 1.3 lakh cases on May 24th. But if you see the mainland China, where this virus was originated, the number of confirmed cases is quite less. And if you see the data, the number of confirmed cases in China is even less than the number of deaths in the United States. You see, on May 24th, India is at number 11, was at number 11 in the list, but it is unfortunately we are rising up the list, whereas China was at number 14
and it is still going down. You see the data after 1 week, that is on May 31st. This data I collected from internet and from good sources. You can see that there are the China, the number of cases are not increasing much. So in a week time, India was at number 8 from number 11, whereas China is at number 17 from number 14. Only 26 cases are increased at China with no single death from COVID-19. Whereas in 1 week, more than 50,000 cases are increased at India with 1,300 deaths. So I think as we are into unlocking more of this prolonged lockdown, we need to be very careful. I think what is important is the strict compliance of citizens with policies such as social distancing, sanitation, and isolation is playing a major role in controlling this pandemic. And I think we need to understand this. And while we are unlocking from this lockdown, we need to be very careful. A recent publication published from USERN, which is a Universal Scientific Education and Research Network, we have shown that national and international cooperation have helped tremendously in curbing the spread of this virus. Very specifically, I think we wanted to highlight that there is a need of a global collaboration to facilitate research in the field of diagnostics, drugs, and vaccines. And now I think most of these participants are chemists. I think we all, as a chemist, we all should be proud that we all contribute in this area of diagnostics, drugs, or vaccines, directly or indirectly. So you are aware about some work about diagnostics. Specifically in terms of drugs in COVID-19, I think Dr. Virana has mentioned about repurposing of drugs, but I just wanted to highlight some of the drugs which are being repurposed. So you know that the drug discovery process is quite lengthy process, it's a slow process, and it takes around 15 years of research to get one molecule into the market. So for COVID-19, it is not possible right now, or it will be of no use to start a discovery from scratch and then get to a new molecule in the market. That's why molecules which have already gone through a rigorous clinical trials and shown to be safe, they are being repurposed. So this is a very interesting approach where you can see that molecules such as baricitinib, which is being used as a treatment for the rheumatoid arthritis, is being repurposed for COVID-19 because it has been tested in cell lines, in animal models, and then it found some good results, and that's why it is being repurposed. Similarly, drugs like lopinavir, as well as other drugs, ritonavir and darunavir, which are HIV protease inhibitors, they are being used for controlling COVID-19. The other drugs such as which target RNA synthesis are also being explored. We know that coronavirus is an RNA virus, and this specific drug like remdesivir, galidesivir, and ribavirin are being used, are being repurposed to be used in COVID-19. There are a few more drugs. The overall story, what I wanted to tell here, is that the organic chemists or synthetic chemists, they should look into these structures and see that how is the chemistry of these molecules can be developed, how some analogs can be developed, or maybe which can be quickly taken further for its possible applications in COVID-19. In terms of vaccine, because my today's talk is not about drug repurposing or how we can get to a new drug, but what I wanted to highlight is about vaccines. There are various kinds of vaccine, and you know that the first one is a whole inactivated vaccine where pathogens are killed through chemical treatment or heat, and that is being used as a vaccine. Second one is live attenuated vaccine where less pathogenic strains of vaccines are being used. Third category of vaccine is the synthetic peptides where most dominant epitopes, so there will be some portion of this proteins can be synthesized and used as an antigen for a specific vaccine. The next one is recombinant subunit vaccine, which can be conjugate vaccine where strong antigen is coupled with some weak antigen. So these are the synthetic peptides which can be used as a recombinant subunit vaccine. Then the next three, which are recombinant viral vectors, recombinant bacterial vectors, or DNA vaccines. These are the new generation vaccines where live replicating viruses that are engineered to carry specific genes. So these are the live replicating viruses which contain only a specific portion of a RNA or a DNA, and they produce a specific antigen or a protein in our body, and then trigger generate immune system against immune response, particularly against those specific proteins. DNA vaccine are the fragments of DNA encoding antigens, and they are also being used as a vaccine. What is the if you see what are the advantages and disadvantages of these vaccines? We have just seen that there are various kinds of vaccines, but if you see there are specific advantages. Like for example, live attenuated vaccines, they induce a long-lived responses, but they are a little bit unsafe. There are adverse effects in immune compromised patients. There are like for example, subunit vaccines. They have a high level of safety because they are highly purified antigens, but they are less immunogenic, and that's why multiple doses are usually required when you want to use subunit vaccines. Like for example, toxides also, booster doses are required, and that's why you know that there are booster doses of vaccines are required, and governments are like for example, polio, you know that there are a lot of booster doses are being given. For conjugate vaccine, you can understand that you have to identify specific epitopes. Then you have to synthetically produce them, and then again you have to combine them with some other antigens. So there will be a huge like chemistry expertise required, facilities required. So therefore they will be highly expensive to produce. DNA vaccines, they are limited to a specific protein antigen. As I said, a small DNA fraction, like a fragment, is being injected, and that's why that DNA is specifically responsible for creating a specific protein, and that's why it is limited to only specific, limited to a protein antigen for its production. The recombinant type of vaccine, they generate strong immune responses, but the problem is the anti-vector immunity. So that has to be taken care. There are definitely advantages and disadvantages in the development of vaccines. It can take years. As I said, for drug discovery, it can take 15 to 20 years. Similarly, for vaccines also, it can take somewhere from 10 to 20 years or maybe 15 years for the development. And because there is a first in laboratory, someone has to show the proof of concept. Then there's a development or manufacturing process to make the stable and and highly purified antigens. And then there will be clinical trials and so on. So that's why this is also lengthy process. But for COVID-19, vaccines has been fast-tracked into humans. And so whenever there is a pandemic, there are special regulations, and some studies are accelerated. So it's not that they are directly tried they are directly brought to the market. So there are some accelerated studies being done for its development in a short time. So let's see what is the current scenario about vaccines for COVID-19. As per May 22nd, that is just last month, WHO, World Health Organization, they produced a data where it shows that there are 10 candidate vaccines which are in clinical evaluation, whereas 114 candidate vaccines are in preclinical evaluation. You can see that in clinical trial, there are 5 different kinds of vaccines: inactivated, non-replicating viral vectors, RNA, DNA, as well as subunit protein antigens. Whereas in preclinical trials, all the variety of vaccines, scientists and researchers have put into the preclinical evaluation. So I think we should thank all the this scientific community, which involve biochemists, microbiologists, and definitely chemists also. So I think we should be thankful to all their research work during this lockdown period. So we should be also proud that there are several Indian institutes and biotech companies are into vaccine development. Specifically, You can know that I think Dr. Viran also mentioned that Oxford's vaccine, they are claiming that it can come in September, but again, there are some results that it did not show good positive results. But then what I wanted to show here that Serum Institute from India is tied up with these efforts to develop and produce vaccines fast. Bharat Biotech has like 2 to 3 programs on with several collaborators. NIV, National Institute of Virology, and Bharat Biotech, they are also like working on the development of vaccines. As of May 11, 2020, Dr. K. Vijay Raghavan have announced that there are around 30 Indian attempts for COVID-19, and he was thankful to all the researchers. What I wanted to highlight is this one. The Sinovac inactivated vaccine is mixed with alum. So what is that alum? Whereas Novavax protein subunit vaccine is adjuvanted with matrix M. So what is this term? Adjuvant. And you know that matrix M is nothing but it is a saponin-based adjuvant. And saponin, as a chemist, you know that they are steroid or triterpenoid glycosides, which occur in many plant species. So that means there are definitely some kind of chemicals or additives to be added in vaccines, and they are termed as adjuvant. So, like for example, for COVID-19, at least the data says that alum, matrix M, Advax, and CPG1018 are being used as vaccine adjuvants. I'm very proud and I feel happy to say that Advax is a discovery of Professor Petrovsky, with whom I have a Spark project, and he is a Spark visiting professor of Punjab University, and he also has a vaccine candidate which is in preclinical evaluation. So overall, you can see that I have shown you all various different kinds of vaccine, and then most of them are adjuvanted. You can see that pertussis, tetanus, then diphtheria, and so on. All the blue dots, these are the adjuvanted vaccine. So what is so special about adjuvants, and why the adjuvants are required in vaccines, and what is vaccination? So before you understand what are adjuvants, let's understand what is vaccination. So the overall goal of vaccination is to generate a strong immune response against the administered antigen, which should be able to provide a long-term protection against the infection. So this is a layman language about what is vaccination. And you know that the early vaccines we generally use, these are the used killed whole organisms, are known to produce several adverse reactions. Those are like many of you might be remembering a TT shot which was given in our school time, and when it was given onto the arm, you know that at least for a week or at least for 3 to 4 days, we were unable to move our arm. So there was so much of swelling and pain in the arm because of the TT shot. But now the new vaccines, which have more defined composition, are mostly highly purified antigens. And the distinct advantage of these new vaccines, the modern vaccines, so-called modern vaccines, is the ease of production, quality control, and safety. So because they are highly pure, so there are distinct advantages. But because of their highly purified nature, they are less immunogenic. And if you remember from my earlier few slides, I mentioned that subunit vaccines, they have to be dosed like more frequently, and booster doses are required. And the adjuvants are the compounds which can be added in this such kind of antigens, they help these poorly immunogenic antigens to induce a strong and long-lasting immune responses. So if you look at the data, there are like hundreds of compounds that are synthetic or a naturally occurring compound. They are known to be adjuvantive, but alum, which is a mineral salt, aluminum hydroxide, was the only inorganic salt, I can say, is approved all over the world. But and for over 60 to 70 years, that was the only adjuvant being used or being approved by FDA. But very recently, MF59, which is oil and water emulsion, is being introduced by Novartis, whereas GSK introduced alpha tocopherol and in combination with oil and water emulsion. Whereas GSK also introduced another kind of adjuvant, which is a monophosphoryl lipid A, and mixed with alum. So it's a combination adjuvant. What are but if you let me look at this more details about what are these adjuvants. You can see that like poly-IC, MDP, which is a muramide dipeptide, then forms adjuvant, again poly-IC is another version, then CPG, which is oligodimnucleotide, MPL, which is monophospholipid, and then these are the RS837, whereas R848, these are the organic small molecules used as a vaccine adjuvants, and then there are some biomacromolecules like flagellin. So what are these molecules? So these are nothing but the immune potentiators. So their role is to induce immune system in the presence of an antigen. So what are the immune potentiators? Let's understand the immune system before that. So infection of the human body by pathogenic microorganisms trigger the immune system. We all know that we have two different arms of the immune system: innate immune system as well as the adaptive immune system. Innate immune system, it acts rapidly. As soon as it come across the infection, it try to call the infection. Unfortunately, it does not create any memory. But while during this fight, you all know that we generally after the infection there is a fever, and then and that is nothing but the innate immune response to the infection, and and then it generates a message to adaptive immune system that wake up. There is a microorganism attack, and then it slowly try to clears the infection and also keeps the memory. This is how. So that our body recognizes innate immune system, and toll-like receptors, they play a role of a bridge between the innate immune system and the adaptive immune system. So immune potentiators are the molecules which activate the toll-like receptors or any other pattern recognition receptor. So kindly remember that I'm going to use this word pattern recognition receptors, and toll-like receptors are one of the pattern recognition receptors. We'll see that in more details. Toll-like receptors are the transmembrane proteins which has two domains: extracellular cellular domain as well as the intracellular cytoplasmic cell. So toll-like receptor, like for example, there are many now toll-like receptors. Crystal structures are known, so which can for chemists it is a good news that we can understand the structures of these like receptors and can develop agonist as well as antagonist for those. Toll-like receptors, they are predominantly expressed on the immune cells. I will explain like how it is useful for development of adjuvants. Their expression profiles vary among tissues and cell types, because of which it becomes useful to target TLR as a good like target for drugs. So far, there are 13 toll-like receptors known, and 10 are found to be functional in humans. For example, you can see that there is a TLR2 which can dimerize, or like by self-dimerizes, or it can dimerize with TLR1 or TLR6, and it recognizes a very specific pattern. So these specific patterns are nothing but the biomacromolecules from the bacteria or fungi. Like for example, TLR2 recognizes lipopeptides. TLR4 like self-dimerizes and recognize LPS, which is lipopolysaccharide, and monophospholipid A, which I mentioned in 2 slides earlier, is nothing but one of the analog of the lipopolysaccharide. TLR5 recognizes bacterial flagellin, whereas TLR11 recognizes profilin of protozoa. There are some TLRs which are expressed onto the cell membrane, whereas there are some TLRs which are present in the endolysosomal compartments. For example, TLR3, 7, 8, and 9, they are present inside here. They recognize, you can see, a double-stranded viral RNA or a single-stranded virus. Now you know that, so there is a system in our innate immune system, receptors like TLR7 and TLR8, which recognizes a single-stranded RNA virus. So now you know that coronavirus is being recognized by TLR7 and 8, and you know that you might have read that there are a lot of cytokine storm, and then because of this virus, it is creating inflammation in the lungs. So it is all through recognition of the viral single-stranded RNA by toll-like receptors 7 and 8. So what is exactly happening here? So toll-like receptors are, as I said, they are predominantly expressed on macrophages and dendritic cells. So they are activated by the specific recognition of PAMs, which is pathogen-associated molecular patterns. These are nothing but the molecules, biomacromolecules from the microbes. Like for example, for COVID-19, for coronavirus, viral RNA is the PAM, which is being recognized by TLS78. Because of the binding of this viral RNA onto the toll-like receptors, there is a maturation of these dendritic cells, and this maturation leads to the formation of a pro-inflammatory cytokines. This induction of pro-inflammatory cytokines then trigger like our adaptive immune system, and they trigger T helper cells, specifically Th1 and Th2 cells, which leads to a cellular as well as humoral immunity. That's how these receptors, they recognize these molecular patterns, and then they induce the adaptive immune system through activation of the innate immune system, and that's how they play they are the bridge between the innate as well as the adaptive immune system. And it was very interesting that while I was working on this topic at the University of Kansas Lawrence, a Nobel Prize was awarded on this area, and this Nobel Prize was given, the half of the Nobel Prize was given for Winkler and Hoffman for their discoveries related to TLRs and activation. Mostly he worked on TLR4, whereas the other half was given to Professor Stillman for his discovery of dendritic cells and their role in adaptive immune system. These discoveries, they have now given so much of information to us, which is now very much useful for the development of several therapeutics as well as vaccine adjuvants. So what is our approach? Like what is my approach currently at Punjab University? So now you know that microbes, they have specific structures. They have cell membrane, they have DNA, ribosome, cytoplasm, they have proteins. So they have several biomacromolecules. To develop vaccine, we need to identify a specific antigen from this whole bacteria, virus, or any microorganism. And what is required is to find out an adjuvant. So what we are trying to do at Punjab University, we are trying to develop novel adjuvants, and these adjuvants are nothing but the mimics of the some of these PAMs, pathogen-associated molecular patterns. What we are trying to do now, once we have identified a good antigen and a good adjuvant, we are trying to assemble them like a microorganism, and so that we can prepare a nano emulsion or a nano assembly of this antigen and adjuvant, which can mimic these microbes. Overall, we are trying to develop new toll-like receptors. We are trying to develop several nano assemblies. We are doing encapsulation, lipidation, and adsorption of these molecules. We are trying to develop self-adjuvanting vaccines. So we are trying to link these two by using simple chemistry, and then we are trying to develop new adjuvant combination. So overall, what we are trying to do on toll-like receptors 7 and 8, let's go ahead and see that. So in toll-like receptors 7 and 8, as an organic chemist, what we have tried to do is we looked at all the literature and found out that there are specific structures in this. Like for example, you can see imiquimod, resiquimod, they are imidazopinolene structures. There is an amine, and there is a nitrogen, and there is a specific groups here. And we have observed that these are the pharmacophores. Without the presence of these specific groups, the molecules are either becoming low active, high active, or they are losing their activity. So there is a specific role of a specific group. So among all of these molecules, we selected to work on imidazopinolene, which has a benzene and butyl group over here, which we call it as a BPIQ. So this we have selected because it is the most active scaffold, imidazopinolene, like for TLR7/8 activity. Many of the derivatives are available commercially, but the most active ones are not commercially available. So our idea was to synthesize the most active analogs and then try to explore them in various different biological platforms. There are, as a chemist, we have looked at that there are low yielding synthetic routes, so it was our challenge to synthesize this molecule on a large scale. So there was a very less like biology was explored on this particular molecule. So as a start, to start with, we took this molecule as a target. In the literature, what we have observed that people have synthesized this molecule. There are 3 different routes we have seen. So, like for example, the first one, this BPIQ was synthesized starting from 2,4-dihydroxyquinoline. In 4 steps, they could reach to this highly functionalized quinoline, where you can see that nitration followed by chlorination and the displacement of the 4-chloro and then reduction. So these are the simple chemistry steps were done, and then this diamine was then cyclized to imidazole by using guanylyl chloride and then dehydration. So the second step was from anthracic acid, where anthracic acid is converted to quinoline. I will explain the chemistry of it in the next slide. And then it was then converted to the desired molecule. The third approach was from Professor David Ferguson, was also an interesting one, where aminomalonitrile was used in a multi-component reaction, was treated with orthoesters and amine to synthesize highly functionalized imidazole, which on then Sandler reaction, like amino was converted to iodo, and then by reaction of like a Suzuki reaction was then converted to this highly functionalized biphenyl moiety and then cyclized to form a final product which has the imidazole quinoline. The advantage of this method is that the authors have they can easily change several groups on these molecules. But there are some disadvantages in this method, that the chemicals used were quite expensive, and to start with at the university, we wanted to look at the simple chemistry. So we selected the second route from anthracic acid, because we wanted to make the exact molecule from the cheapest route. So we started with anthracic acid, but anthracic acid, being a drug precursor, is banned and is not available to purchase in India. That's why we've chosen a simple route, which is mentioned in Bhurao. Our BSc student used to synthesize this molecule from thalidomide to anthracic acid, and then thalidomide was very nicely available in our BSc labs. Bromine was available. So by using this Hoffman rearrangement reaction, we synthesized anthracic acid on gram scale. Anthracic acid was then treated with 2 equivalents of nitromethane, where it forms methazoic acid, and then reacts with anthracic acid to form this nitro intermediate, which was then cyclized using acetic anhydride to form a nitroquinolines. The 4-hydroxy was then activated and then was displaced by benzylamine. Interesting what we have done here to the optimization of this nitro reduction, where only 1% of charcoal was used here, and by using a balloon hydrogen pressure. We have also optimized this cyclization process, where instead of like auxiliary chloride or valunyl chloride or the related acid, we have used trimethyl orthovalerate, and the reaction goes very clean, and we got the desired product. So after then the next step was the insertion of amine, for which we did an oxidation followed by treatment with the benzoyl isocyanate to form the desired amine. Benzoyl isocyanate also is quite expensive from Sigma, so we have synthesized this lab from benzamide and reacted with auxiliary chloride, and then we use in situ to form the desired product. This work we have recently published in Human Vaccines. So what we have done, so now you can see that this is a imiquimod, which you can see is active in TLR7, the green line, and you don't see that it's not active in TLR8. So the only, whereas its analog, vesicimod, it is more active analog of imiquimod, and it's active in TLR7 as well as it's active in TLR8. So now for a chemist, you can see that a very small change. Introduction of one of the hydroxyl has increased its activity, as well as one introduction of the chain here, maybe I can say 4-atom chain of the oxygen. It has increased the activity, and it has also increased, given its activity. And now once we change these groups, we can see that benzyl molecule, it is a more active TLR7 agonist, but it does not have TLR8 active. So it is a TLR7 specific, more active analog of imiquimod. We have tested this molecule as an adjuvant by using recombinant hemoglobin as an like antigen, and we have seen that more than imiquimod, it boosts IgG as well as IgG2 like immune responses, and so this work we have published, as I said, in human vaccines early this year. Interestingly, what we have done, we have tried to explore the application of this molecule in malaria vaccines. We have isolated a blood stage fraction of like 10,000 G. This work was done in collaboration with Professor Mabagai from Department of Virology, Punjab University. She's no more, but Professor Sukhdeep Kaur is now helping us to take this work ahead. So here you can see that when we have given like this group 2, the infected control, when we given the like on the infection, the animals started dying. Whereas in group 3, where we are given fraction plus BBIQ or compound, so you can see that these animals, they started growing the infection, but after some time they completely cleared the infection, and then all the animals survived, no death. So this combination worked well as a malaria vaccine. You see, when we given BBIQ alone, only our compound alone, you can see that the infection didn't started immediately. At the start, infection was less, and then out slowly the infection rise. So as a chemist, you know that this is organic small molecule, so it is inducing the immune system, and that's why the mice they fight against the infection at the early stage. But then as this small molecule, it was excreted out from the system, and that's why after some time the infection rise. So this work we have communicated to scientific reports early this month. But based on this observation here, that these molecules they induce immune system, and then animals can fight the infection, we have taken this molecule and we have tried to develop a chemoimmunotherapy where we have mixed this molecule with chloroquine, and we have observed that animals could survive up to day 23 because this molecule was inducing the immune system, and when we mix with chloroquine, and then when we are using a suboptimal dose after 23 days, when this both the molecules are less in the system, then the infection is growing. So this combination can be used as a chemoimmunotherapy. So if we can change the dose of this agonist as well as chloroquine, we feel that we can get to a good combination therapy for malaria. Recently, we have also used this study this molecule for leishmania, where we observed that it's acting as a pro-oxidant or pro-inflammatory agent. So that work I will, because of the lack of time, I will not explain here. But what I wanted to show here more chemistry, like while we were synthesizing like several analogs of imidazopinolene, we designed like benzimidazopinoxaline, and then synthesized a library around it. And we observed that this molecule unfortunately lost. There was no TLR7 activity, but then we interestingly observed like a good anti-cancer compounds through this library. This work was done by Rahul and Ravinder, and the activity was done at NCI 60, National Cancer Institute 60 at USA. So in terms of like now we wanted to, we asked a question whether can we develop a sustained release formulation, because we have seen that as a vaccine adjuvant, we want to give these molecules like they should be released into the system slowly. So because you know that this TLR 7-8 are inside, and the molecule has to cross this membrane. And in my earlier work during my PhD, we have shown that molecules which have antibiotic nature, hydrophobic nature, they can disrupt the membranes. That's why we have tried dimers which has a hydrophobic phase and a hydrophobic phase and a hydrophilic phase. To make a dimer, they can make a sandwich kind of conformation, and which can disrupt the membranes. Very recently, one of my students, Poonam, she synthesized many of these dimers, and then we identified a specific dimer which, when when combined with the different antibiotics, you can see that it has increased the activity of amic acid to 67-fold. And you can now see that amic acid is a highly polar molecule, and it can fit into the polar cavity. This work last year we published, in 2018 we published in Journal of Medicine. So now we have identified a molecule, and now we are trying to develop a combination with tailor agonist, so that tailor agonist can be given to can be targeted to the receptor inside the cell. Poonam has also developed another antimicrobial peptides, where she has linked some different amino acids to steroids, and we observed that interesting biological activity. So these are the amphipathic steroids we have developed, and we are trying to now make a combination therapy with TLR agonist. She has done an extensive literature survey of possible applications of steroid amino acid conjugates, and which we have published early this year in European Journal of Chemistry. So overall, what I wanted to do, because we are also developing the combination adjuvants, so we are trying to see if we can use TLR agonist, TLR2 agonist, as well as TLR7 agonist together. If you look at the TLR2 agonist, the known TLR2 agonist, PAN2CSK4, if you look at the structure, it has a hydrophilic head as well as a hydrophobic tail with a spacer of a dipeptide. This molecule is highly expensive. Only 1 milligram, it is commercially available from in 1 milligram costs around 25,000. But then it can form micelles. So our idea was to make the micelles and then mix them with the TLR7 agonist. So, Poonam and Ashprit, they took this challenge. So they synthesized PAM2CSK4 because it was not possible to buy so much of PAM2CSK4 required for this formulation studies. They synthesized this in 20 steps. So it was like a retrosynthesis approach I'm showing here. Ashprit synthesized PAM2CS component, Poonam synthesized tetrapeptide, and then from they use the synthone this isopropylene glycerol, which is commercially available. And also from this amino acid serine. Oral synthetic process involved a typical simple synthetic steps where we have used this mixed anhydride strategy to make an ester, and then there was a deproduction was done by by using piperidine, and then it was coupled to another protected lysine, and then step by step we built this tetralysine. While Ashprit started the L-cysteine, and then she built the PAM-CS part, again by following a typical synthetic chemistry. Because of the lack of time, I will not go through each and every step. Then finally, after total 20 steps, they synthesized PAM2CSK4, which was published 2 years back. So far, from in TLR2, from a very complex molecule, this slide I specifically wanted to show it to all the students that by doing a typical a simple synthetic chemistry, we can convert complex molecule to a simple structures. Even PAM2CS was then simplified to PAM-CS, and then we have increased its solidity by adding several polar groups into it. So a medicinal chemist can very cleverly make these changes in the structures and can develop very interesting molecules. So recently, Ashprit has done now a systematic structure activity relationship studies where we have done a lot of changes. This work is just communicated to OBC. We have done some changes in the civil chemistry and some changes over here. And you can see that we have got, this is the PAM2CSK4 is here. And so we have got molecules which are active. You can see the activities from a micromolar range to a nanomolar or to a picomolar range. So we have some molecules which are quite active. This has been done by a SAR, structure activity relationship studies. We are also trying to develop formulations of it. So my student Jyoti, in collaboration with Professor S. K. Mehta, we have recently developed niosomes where we have synthesized a niosome by using protonexamide, PEG, and water. And now we have tried to mix up BBIQ and B1 and B, which is a IDO inhibitor. Again, we are very keenly working on to develop novel IDO inhibitors. So here we have developed a combination of IDO inhibitor as well as TLR agonist. So overall, in my today's talk, I have told you that what is the vaccine status for COVID-19. I have also told you about how total is a new target for development of vaccine as well as chemoimmunotherapy. So I have shown you our own work for development of vaccine adjuvant as well as malaria chemoimmunotherapy. I have also shown you like how we have synthesized time to CC4 in 20 steps, and then also a little bit shown you about the application of antibiotic antimicrobial steroids. Thank you. So this work was all possible because of the extensive collaboration. I'm thankful to Punjab University for providing a very good infrastructure here at our chemistry department. Very good support from department. And then I'm thankful to our collaborators, especially Professor Petrovsky for helping in the vaccine development work, and then all of them for malaria, leishmania, anti-cancer studies. I'm thankful to all the funding agencies for generous support, because without which it is not possible to do any research. So I'm thankful to all my students, because whatever I presented is all the efforts of the students. I think today I talked about work of Ashpit of Kela 2. Then a little bit I told you about work of Rahul and Ravinder on anti-cancer agents. I also told you about some work of Dipender, who worked on Kela 7-8, and also told some work about Poonam and Jyoti. So thank you all for listening to. So this is Professor Petrovsky, who visited us on a SPARK program, and he has a vaccine candidate for COVID-19, and we are trying to see how we can help him for its further development. So thank you all. Hello?
Vipin sir.
Yes, sir.
Yeah, so if the participants have some questions, I can take questions.
Hello.
Yeah, any questions, sir?
Are question? Nice, sir.
Okay, I think participants are tired. They might be hungry. It's time for a lunch break.
Right. Vipin sir.
Yes, sir. I am Dr. Vipin Shinde. Herein I am giving the vote of thanks. Dr. Deepak Salunke has an excellent presentation. It is a very important presentation due to, since this is the pandemic situation of COVID-19. He fluently explained the vaccine status of COVID-19. Further, he explained the role of adjuvants along with the antigens. And again, he explained the toll-like receptors as new target for the vaccine. He explained this role of synthetic organic chemistry for the development of vaccine. He also explained the importance of BBIQ as an important adjuvant. This presentation is very important for the young generation minds, because today is the era of, I think, era of vaccine preparation. Thank you so much, sir, for the excellent presentation for the new researcher, sir. Over to Padwa, sir. Padwa, sir. Hello. Hello, Padwa, sir.
Sir, बोला ना.
Sir. चला ना?
Yes, sir. Sir, okay. Now this is the time for lunch break. After lunch break, we have the oral presentation. We can display this oral presentation on YouTube link. Thank you for attending all session. Thank you. We are sending feedback links, madam, sir. During yes. We are sending feedback link in few minutes. लागली राव नाही लागलं. पाहिजे ना अगं सर बोल ना.
हॅलो वधवा सर.
हॅलो सर.
हा बिपिन बोलतोय सर.
हॅलो. हा बिपिन ओके ओके.
हॅलो.
हा बिपिन आता आपण पेपर प्रेझेंटेशन घेणार आहे.
हा ते पेपर प्रेझेंटेशन घेऊया का पोस्टर.
पेपर तयार झालंय सगळं. भागवत सर अपलोड करतील.
बरं ठीक आहे ठीक आहे ठीक आहे ठीक आहे. आता लंच ब्रेक आहे ना.
हो लंच ब्रेक.
ठीक आहे ओके.